I'm very happy to open also this year the second edition of this conference, which has several points of interest, also from a strategical point of view for our university and for the international cooperation that you are able to set up. First of all, I want to thank all the organizers and thank for all the speakers that are connected with us. We restarted doing something in presence, but not yet fully. And I hope that the third edition will be full in presence here in Brescia or abroad. And thank you for the guests that are here from other universities, the Polytechnic of Turin, and all the members of the staff of our two departments involved in this conference. The first point of interest is that this is a multidisciplinary conference for us, and I think also for the speakers and the participants abroad, that put together different competencies. And this, I think, is very important in the light of the 17 Sustainable Development Goals that we want to reach within 2030. We are already at the midterm because they were set up in 2015. So already 7 years has gone and we have only 8 years left to reach these 17 multidisciplinary goals, because in those goals there are different competencies and topics that are called to action. And especially when we speak about food and fighting against poverty and providing food for everybody on the planet. The second point is this worldwide perspective. As university, we are rooted in the place where we are, like in this province, but the mind is open to all the world. And the problem of food, providing food and safe food, secure food, is a problem that is planetary problem. And in this town, we have a very strong history of cooperation, international cooperation, also with the underdeveloped countries. And so I think that this conference is well rooted in the experience of the town of Brescia. If I may say, for the first time, the law in 1966 was approved thanks to a parliament member of Brescia. It was a law, very important one, that allowed people, young people, to spend some of time abroad for a cooperation project, 2 years, and that was in alternative to the military service. It was very advanced regulation, 1966, and really thousands of young people had the experience to go abroad for cooperation project. Also in these fields, starting from, as I said, a member of the parliament that was born in the province of Brescia. A third point of interest, apart from this disciplinary topics where I'm not really fully competent, is that in Brescia we have an institute, a research institute that is called Zooprophylactic Research Institute, that is celebrating its first century anniversary. Yesterday we celebrated it. And so we have also the biggest institute in Italy that makes research on these problems, especially on the animal diseases, but they have been, during the pandemic, really a great support to the fight or to detecting of the virus. So I think we have also to be proud in this town to have the biggest research institute, public research institute about those topics, and that is working competent on 2 regions, Lombardy and Emilia-Romagna, but based in Brescia, placed in Brescia. Well, I am an environmental engineer, so I know what it is an ecosystem. And I think when we speak about One Health, or the relation between the health of plants and the health of well-being, we are in the same perspective of dealing with ecosystems. So in the ecosystem, we are all linked. And you know that the main formula that regulates the life of ecosystem is equilibrium. It's not the growth, it is equilibrium. So we have to be in equilibrium with the other components of the ecosystem. That is the challenge for every one of us that is dealing with this kind of problem. And so when you speak about One Health, that is a new expression to say, I think, the relation in ecosystems, so we are all relating one to another. I think that you are really in the core of the problem that we are facing today when we speak about environmental problems, so that we do not consider ecosystem as one whole, as one entire entity where we have relation, you know, energetical relation, exchange of materials and exchange of relation inside the ecosystem. And this is, I think, one of the new paradigm we have to approve, we have to assume that is not in opposition to the traditional paradigms, but it is the evolution. It is the evolution of the traditional economical, neoliberal economical paradigm, that is growth is the goal, not the equilibrium, and the technological paradigm, that means everything is possible from a technological point of view is good, which is not true. So I think that even if the conference is very well rooted in some of the topics of the ecological crisis, I think that you are perfectly in the mood of what we have to together really advance in research and politics, also in policies, also to get as close as possible to the 17 Sustainable Development Goals. The last point is about the international crisis we are living. I've been rector with many crises, the COVID pandemic, the Afghanistan crisis, the Ukrainian crisis. I'm starting thinking that is my problem, so, but I will soon finish. So crisis will probably be finished, will be over. But the Ukrainian crisis is also lightening another point, that is that of food. We are 7.8 billion inhabitants on the planet. The forecast is 10.9 billion at the end of the century. So we need more food, and we are devastating a country that is producing a great amount of cereals that we all use. So first problem is human lives that we are losing, but I think the second one is the ecological problem, also because of the war in Ukraine. That is a country that is double, twice the surface of Italy, and it is really, you know, the producer of a lot of cereals we are importing. And so why we do not reflect also that the ecosystem crisis is the ecosystem, sorry, is a crisis of the relation between man, plants, animals, but also man and man, women and women. And in this kind of crisis that we could avoid, because we have to do a lot of research to solve the problem of the ecosystem. Many things we still do not know, but we know very well that wars are a catastrophe in any sense, in any way, in any latitude and in any time in history. There is no positive result out of a war. We are hosting some of Ukrainian students and researchers also in our university, as in all universities in Italy, and we will continue to do that because we really think that we are one health and one planet together. Thanks to all the colleagues, because we have different competencies at the table and in the audience, and I think in the speakers, that goes from the medical school, from the biological school to the engineering. And this is a very good example of how we can cooperate to solve the problem. The main problem we have in front of us, that is the ecological crisis. Thank you very much. Have a nice day of work.
Thank you, Rector. Thank you, Professor Tira, for the interesting points you focused in your talk. I'm now giving the word to Dr. Hing-Huan Xia, the Director of the Plant Production and Protection Division of the Food and Agricultural Organization of the United Nations. Good morning.
Okay, thank you, facilitator. Dear Professor Gabi, members of organizing committee, ladies and gentlemen, good morning from Rome. I'm very pleased to present some opening remarks at this important event with the theme from plant health to community health. First, I would like to express my sincere congratulations to all of you for organizing the second edition of this webinar series. And I also want to express my high appreciation to all organizers for your kind invitation. I'm very glad FAO and the University of Brescia have this opportunity to collaborate again on this issue related to plant health and food security. There is no life without plants. Also, plants provide us with the food we eat, the oxygen we breathe. There is no food security without plant health, because 40% of our food crop lost due to the damage by plant pests and disease every year. As you may know, FAO is implementing its strategic framework 2022 to 2031. This framework seeks to support the 2030 agenda through transformation towards more efficient, inclusive, resilient, and sustainable agri-food system for better production, better nutrition, better environment, and better life, leaving no one behind. Within this framework, one of the key program area is innovation for sustainable agricultural production. This project is led by FAO Plant Production and Protection, we call it NSP, our division. NSP's mission is to enable transformation to more efficient, inclusive, resilient, and sustainable plant production and protection through two-year strategy, optimization and minimization. This means to optimize all positive aspects in the cropping production system, better structure, better functionality, and better service, and to minimize aspect in crop production system. Less losses of crop yield and biodiversity, less residual risk of highly hazardous pesticides, and less contamination of agricultural chemical. With regard to production, and this, we are leading the implementation of FAO Global Action Green Development of Special Agriculture Product. One country, one priority product, or we call it OCOP. It's a very big initiative from FAO to support nowadays production. There's two words that's important. One, development. Another one, special agriculture products. There are four characteristics of inclusive of this OCOP initiative. Country inclusive. All the country can be engaged. Inclusive, including animal products, plant products, animal products. Value chain engaged, inclusive. This means not only to production, also storage, processing, and marketing. And also stakeholder, inclusive, government, academia, civil society, private sector to be engaged. And there's a four core areas for this global action. We call the four green technology, green production technology, green storage technology, green processing technology, and green marketing technology. So, taking this opportunity, I would like also make an announcement that FAO is organizing global conference on sustainable plant production with the themes innovation, efficiency, and resilience. This conference is the first time in FAO history organized it. the first ever in history, we can see, will take place on 2 to 4 November this year, 2 to 4 November this year. And also we hybrid many men in Rome and also virtually globally. So you are warmly welcome to the first ever conference on global sustainable plant production. With regard to protection, I feel we are leading the plant health management to support the global One Health initiative. So we are mainly focused on 3 areas. Number 1, foster development and implementation of international standard on phytosanitary measures to protecting global plant resource, while at the same time facilitating the safe trade. Second, strengthening sustainable pest management through promotion of green plant protection and digital plant protection. And once again, plant protection and digital plant protection. And number 3, create enabling surrounding for plant health in soil health, seed health, and pollinator health. At present, we are leading 3 global programs or global action on plant health management. Number 1 is IPPC e-phyto solution. This is a digital trade. FEO global action on fall armyworm control, and then FEO emergency control of desert locust. As you know, in 2020, we FEO successfully delivered the International Year of Plant Health, IYPH, which was once in a lifetime opportunity to raise global awareness of how protecting plant health can help and end anger, reduce poverty, protect the environment, boost economic development. Just 2 days ago, May 12th, we successfully celebrated the ever first International Day of Plant Health. We got participants 1,400 and from 144 countries. It's very successful. The objective of the celebration of Plant Health Day is to continue raise awareness of the public, policymaker, academia, and private sector in their key role in promoting plant health management. So contribution to achieving UN SDGs. Indeed, NSP has a big task. We cannot do everything by ourselves. Well, greatly depending upon the strong support from over 200 NSP. I'm sure most of them, you are also with us. This covered 80% of our partners are science and technology based. It is clear that the academia and the research institution play a key role in delivering science solutions for green and digital technology in phytosanitary, pest, and pesticide management. I'm very glad today to know that the thematic topic of this conference is on biotechnology and functional food. That this is really front chair topic and a very hot topic nowadays, which is also closely related to NSP mission. In conclusion, I hope that this initiative will continue raising awareness of the importance of plant health among policymakers, civil society, academia, and the private sector to engage proactively with international institutions like FAO, among others, to deliver necessary change towards sustainable food system, preserve of our environment, and the facilitation of a safe trade. I wish you all a very successful and fruitful debate. Thank you. Over to you, Chair.
Thank you, Dr. Shah. I think our university and FAO align in the One Health vision and the crucial role of plant health. And now we continue with the keynote lecture session.
Good morning, everyone. I have the pleasure to present Kiladas Shetty, Professor of Plant Science at the North Dakota State University. He's currently the Associate Vice President of International Partnership and Collaboration and Founding Director of Global Institute of Science Food. He's one of the main experts in agriculture and food science. Carry out research is focused on innovative strategies for global food security and health in a sustainable environment. The title of his presentation is Advancing Climate Resilient Food System for Human Health. Please, Kalidas, the stage is yours.
And I must thank the organizers at the University of Brescia for kindly inviting me to this presentation to give this address. And what I'll be talking about is how, as we move forward with climate challenges, how we build a holistic systems-based approach to our food systems and food security, and the foundation of plant systems for building very climate resilient food systems that also takes care of challenges of climate resilience of our agriculture and food, but also how we need to integrate human health countermeasures to this paradigm. So with that in mind, my presentation will focus initially on concepts. How do we build these systems rational to then advance the solutions to these challenges? So if you look at the global innovation economy today, we are at a very interesting point in Earth's history where anthropogenic-based decline is potentially possible because the environment in which we live, that's very fragile, has lost nearly 1/3 of the known biological systems in the last 100 years. And as we move forward beyond the atomic, biomolecular, and computer revolution, we need to look at a more systems approach of how to balance this environment and the biological system, and how this humanity needs to be a much more compassionate species, and also align the technological advances we are making. So in all, these are the 4 key pillars of this innovation economy, where a systems-based approach is essential. And what the systems-based approach is, I will point out in the next few slides. So I see these 4 areas: the environment, biological systems, the human ability to function within them, and the machine tools we build as the key 4 pillars of the global innovation economy in the 21st century. So if you look at the global challenges today, we are a population of about 7.5 billion people. By 2050, we'll be about 9 billion people, with more people living longer. But at the same time, since 2000, we have a new challenge where while we have 1 billion people who are still hungry and need macronutrients and micronutrients, we have nearly 2 billion people of 7.4 billion who are moving more towards an excess calorie and refined calorie burden that's seriously causing a major comorbidity link non-communicable chronic diseases. So this global obesity crisis is a very fundamental foundation to even how we face pandemics, infectious pandemics like the corona, where the comorbidity associated with the obesity crisis makes us more susceptible. So while we address these new challenges of food health and wellness, the technological and economic evolution that's coming from the overall life science advances and information technology is going to impact the quality of life. And more importantly, the environmental consequences of all our action and how sustainability has to be a foundation of these advances. And the most important of that sustainability challenge is how we move away from carbon, fossil fuels, and coal, which is 60% of our energy basket, to a more decentralized renewable platform where we do not build the carbon burden. Great.
This meeting is being recorded.
Thank you to organizers at the University of Brescia for kindly inviting me to this conference on plant health to community health. And I want to take this opportunity to present a few foundational concepts on advancing climate resilient food system for human health. And the rationale behind this is, as we face climate change and try to build our food system towards resilience, we also need to address both climate resilient agriculture and food systems and food security, but also how to integrate human health challenges into these solutions. With this in mind, as we look at the future, we have built our scientific and innovation foundation from the technologies of the 20th century, as we understood the atom and the biomolecular revolution from the DNA and onwards into cellular life, and ultimately end of last century, the computer innovation. But as we go forward, we are in a more systems-based approach where all these knowledges are being integrated. And as we look at the big, larger challenges, we face serious environmental decline, not just climate change, but the biological species within that. 1/3 have been lost within the last 100 years. And further, how do we build the sustainability with human being the key anthropogenic burden on the planet, and how we work with our innovation to build better machines, but at the same time balance the environmental and biological system decline. So as we move forward in this century, the innovation economy and sustainability challenges will be primary. And these will be the 4 key pillars of the human innovation economy. How do we balance the environment, the biological species decline, and how do we sort of put a balance on that? And more importantly, our human function, our human health, and more importantly, human species, does it have a compassionate link to the environment and biological systems, and align the biological, non-biological machinery that we build, or the tools that we build from the non-biological realm to help with our living and sustainability challenges. So these are the 4 key areas I see as the key innovation drivers. So as we move forward, if we look today, we are a population of about 9 billion people. And we have population about 7.5 billion. And by end of 2050, we'll be about 9 billion. And suddenly since 2000, we have a new challenge where we not only have about 1 billion people hungry, but we have about 2 billion people having too many macronutrients burden, leading to non-communicable chronic diseases, largely from the global obesity crisis. And while these non-communicable disease burden emerge, they also feed into our higher susceptibility to infectious disease challenges like the COVID pandemic we now have, where you could see a link between non-communicable chronic disease and where the susceptibility burden is higher. Of course, the data is still emerging. And as we see as food health and wellness challenges are technological advances primarily driven in the life science arena and also information technology shaping the quality of life, we have to seriously look at environmental consequences of our action. And more importantly, how do we move away from carbon towards a more decentralized renewable platform, which also then has other key factors, water being the foundational element of life. How do we balance all of these challenges into a more greener, carbon neutral future? So for this, we need an integrated systems approach where all of these emerging challenges are founded in common core principles. So this complex biological system and the entity on Earth and the emerging properties that are irreducible have control points. So only when we understand these control points that has consequences on food and health and agriculture, environment, only then can we have a better hypothesis to sort of address all of the issues, not based on the components of the system, but based on the control points that shape the overall system in which all of these critical challenges are embedded. So only when we look at the control points and see the emerging properties being governed by that, can we now have a more rigorous hypothesis-based logic and a more systems-based approach to solve these problems that are interconnected. And to build this systems rationale, we need to look at the Earth's history and evolutionary life from which agriculture and economic systems were built. And there are many points in Earth's history, but if I look at some of the things that I've been biased to, as being one, where is the foundation of water on this planet? Because only when water is split by solar energy can carbon be hydrated and create the first molecule, carbohydrate. And so this amazing ability of water to exist in three phases, solid, liquid, and vapor, provides the foundational part of life. And more importantly, how did then this lead towards the early oxygenation by prokaryotic photosynthetic bacteria, which ultimately led to its rapid decline around just after around 300 million years after between 2.4 to 1 billion when they evolved initially. So following this decline, subsequently there was a billion years later, a eukaryotic life that emerged around 900 million or around a billion years after the early prokaryotic emergence. So this eukaryotic oxygen-based life essentially emerged from engulfing of the earlier prokaryotic system, then became the dominant species on the planet. So this oxygen-based life, if you further look at it, you had another major event where the mammals emerged, which is our ancestors. So mammals emerging and dominating the photosynthetic and oxygen-rich world, which then ultimately gave rise to the hominids walking on 2 feet, including us, Homo sapiens sapiens, largely shaped what is happening in this anthropogenic potential decline, which is the 6th decline. We have had earlier 5 declines of Earth, but here's the 6th extinction, largely defined by the Homo sapiens sapiens. And to look at this last big change is the last ice age. And since that 2000 years of the last ice age is when our agriculture domestication happened, first in Neolithic communities around 10,000 years ago, but ultimately domestication in the river valley civilizations, all the way from Africa to Eurasia, wherever the Homo sapiens sapiens migrated. And ultimately a big factor of the current food ecology and agriculture ecology we see is the European mercantilism 400, 500 years ago, which resulted in mass global exchange of crops and animals from the origins of diversity, and that shaped the current agriculture. So these foundations give life to sort of give an understanding of how these emergence of biological system happen on the planet and how we now have this agriculture food ecology, including the relevance of plant health that we want to talk about today. And therefore, if you look at this web that emerges, we need to look at the common control point that shaped this and how complex organization emerged and the diversity emerged. So with this evolutionary systems-based approach in an oxygen-rich world where water being split by plants that allows the hydration or carbon being hydrated, giving the first major macromolecule, is the foundation of our food system that we see today. And we now have a comprehensive function around these plant systems. So using this plant systems-based approach, we need to look at how these basic molecules, carbon, dioxide, water, nitrogen, assemble the food, but also how oxygen-based life, the eukaryotes that emerged 1 billion years ago, are shaping the critical function, which including human species. And so it is that oxygen control point is where all of the breakdowns occur, and where the carbon assimilation from carbon photosynthetic process where the assembly takes place, and then you have the food web emerge from it. So if you take this systems-based approach, we can find the common control points of the overall system. And this is what we have today. We still have this carbon energy, but unfortunately, this carbon is burdened by the fossil fuel and petroleum-based agriculture we have developed, or industrialization we have that has emerged. And this has consequences on the global warming that we see. But also from agriculture and refrigeration system, we have been depleting the ozone, which is sort of the protector of the planet. And that's sort of being stopped at this moment. And this overarching protective layer and changes is what is sustaining the carbon fixation and plant systems approach, from which we develop our macronutrients and micronutrients under this control point where the eukaryotes and prokaryotes work with each other. So that is a cellular system on which our planet depends on, but largely driven by a photosynthetic carbon fixation model. So with this, we have shaped our agriculture, food security, health, energy, and all the disturbances that you see and the consequences of that, food safety, sustainability challenges, the economic system that we have built that is not sustainable, whether it's agriculture production or overuse of fossil fuels, a range of human activities that have emerged that has pressured sustainability and therefore our human resilience. So if we go deeper into this rationale, we need to go back to our biological balance. And therefore, if we see where are the breakdowns happening, the key breakdown is happening at the terrestrial level, where the foundation of our plant food system is. So this soil, terrestrial environment needs to go back to the interaction of the microbial system that has nourished it and developed this soil system, from which our food systems emerged. And it's from this food system is where we get our human nourishment, whether we eat plants or whether we eat animals that feed on the plants. So this integrated approach all the way from soil health to human health has to be integrated rigorously with the interface of the eukaryotic and prokaryotic biology. So this equilibrium shapes our food chain and sustainability of our food chain, and therefore our human health. And I will elaborate on this further. And so if you look at that, then diversity and resilience is the foundation to resilience. So the most critical food and nutritional security solutions come from only when there's resilience. And the resilience comes from diversity of the eukaryotic and prokaryotic balance, from food crop diversity, ecosystem diversity, and then the community diversity from which our different ancestors from different parts of the world developed these solutions from the same ecological platform or similar logical ecological platform from which many different diversities emerged, food diversity, cultural diversity. And therefore, if we take this diversity resilience approach, we can better address the food and nutritional solutions that we are facing. And therefore, agriculture and food system has to be balanced with the ecosystem, and therefore our community system and community resilience from which the nutrition and public health solutions can emerge. And continuing with this diversity resilience approach, we at North Dakota State University within the food security realm, we are looking at 4 major challenges. One, how do we continue to feed the 1 billion people that are still hungry and not getting macronutrients and micronutrients? Number 2, how do we deal with this emerging new non-communicable chronic disease, preventable non-communicable chronic disease like diabetes and its consequences from too much of macronutrients? And third, how do we address both these challenges with climate resilience in mind, where the carbon burden is affecting all our agriculture production? And number 4, many of the antidotes for preventing non-communicable chronic disease or enriching our diet with minerals and vitamins come from fruits and vegetables, so which are highly perishable. And how do we prevent preservation of these foods, of these fresh foods and vegetables? So these are the challenges from which we want to address, where as we face at the end of the century 11 billion people, with current projected to be 9 billion by 2050, what we currently see is we have enough calories for 12 billion people when we have only 7.4 billion. And therefore, this excess calorie burden or preventable non-communicable chronic disease is the most rapid health burden, a growing health burden we face. So with that in mind, you can see the impact of this already emerging in the underlying mystery of COVID susceptibility, where comorbidity linked failure of the food system is very evident and across different communities. So food health and wellness issues and excess calorie burden and the link to obesity, NCD crisis, and comorbidity crisis, and how that affects infectious disease burden needs to be looked at closely. And currently, I have no time to go into this, but data is emerging that there is a link between NCD crisis, comorbidity burden, and higher potential susceptibility to infectious disease, including COVID. So if you look at then the failure of the food system, we have built it on a macronutrient-micronutrient balance, which is valid. But what we are seeing is, while the carbon is a key point of fixation with water being split and carbon being assimilated and nitrogen being added to get amino acids and proteins, is really the oxygen is the critical control point for our energy function. And therefore, if we can put the redox biology protective function or protecting of the mitochondria from the evolutionary strategies of plants or phytochemicals, then we have a much more complete food system where oxygen protection built from the rationale of how plants deal with it, then enriching our diet with more plants and plant-based foods would help to provide solutions to this oxygen-linked malfunction. And not only that, the critical components of fiber and all the other protective elements that the plants have, also very critical to feed our good microbiome. So this oxygen metabolic function breakdown and the microbiome breakdown are the two major breakdowns of our current food system that we need to correct. And therefore, we need a more diverse whole food profile. And this is not complicated, because we just see how nature is working. It's working with this ancient prokaryotes, still working closely with the eukaryotes that's emerged a billion years ago. And then this close interaction of the prokaryotic eukaryotic beneficial interaction should be put into our food system all the way from soil health to human health. So with this systems-based approach, integrating both the role of oxygen biology and its deleterious effect if we do not have optimal diet, is the critical breakdown we need to correct. And therefore, a system-based approach that builds the microbiome platform all the way from soil health to plant health, the human microbiome and the human ecosystem, all nourished by the beneficial microbiome, is very critical as we move forward. And this is particularly relevant as we build our retune our diet to a healthy platform with higher fruits and vegetable whole grain approach, where support of the probiotics or beneficial microbiome supports the overall metabolism, better immunity, better chronic disease management, all from the foundation of good, improved gut health. So this microbiome-based solution to our food system is necessary because it's the foundation for our good health. And the other one is, at the end of the day, it's the mitochondria is what's shaping the higher eukaryotes at the energy function. And so this mitochondrial protection is very much essential because it's the oxygen that's a terminal electron acceptor from which if you see the mitochondrial function is where the proton motor force ultimately couples with the generation of water. And the stability of this water generation depends on how reactive or not reactive oxygen is. So the overreaction of the oxygen is very well countered through evolution by not only the antioxidant enzyme mechanisms, but also in plants, a lot of the protective phytochemicals that have come to counter it. So more of this protective foundation to protect our mitochondria, and especially the mitochondria function to driving the pentose phosphate pathway, which is the anabolic protective mechanism, where the anabolic protective mechanisms emerge. So balancing this important mitochondria and cytosolic pentose phosphate pathway is an important balance that is needed as we sort of retune the chemopreventive benefits of phytochemicals and plant foods. So with this approach, fine-tuning oxygen biology at mitochondria, and especially with the evolution of plant life on land, terrestrial world for 500 million years ago, needs to be looked carefully as we look at disease sensitivity that's made worse in a calorie-rich diet with reduced phytochemicals. And also this logic between fructose-rich foods like high fructose carbon enriched foods versus more starch-driven whole food and fiber-based rich foods. How are they all fine-tuned to the combinatorial profiles, especially phytochemical profile that balances the oxygen function? So this antioxidant coupling, or the function of oxygen biology, is very central, because as we have these high refined calorie burdens and environmental pollutants further complicating it, if you see all the major disease burdens we face today from inexpensive refined sugars and the obesity burden, starting with diabetes, cardiovascular disease, cancer, arthritis, and ultimately consequences on aging. So this phytochemical oxygen protection module is not new. Plants have always used it in both stress adaptation and second reproductive function, which are both survival mechanisms. In all of these survival mechanisms, the combinatorial biochemistry of the redox balance is very central, whether it's antioxidant response, signals for development, seed vigor response, disease response, and fighting pathogens. All of these beneficial interactions of plant have a close link to the redox coupling, and the redox coupling then to the carbon assimilation. So this carbon assimilation redox balance is very central. And if we can pick this up from diversity of ethnic foods from around the world and the species that we have brought into our cultural framework, into our gastronomic framework, then we have a foundation to a good dietary balance. And the exciting thing about plants is they have many diversity of chemicals to doing the same protective function. And therefore, as we sort of tap into this diversity, we get common control points and common logic of many compounds. For example, rosmarinic acid is a biphenolic compound which has a lot of redox regulating protective function. But then the same related function, you can see it in curcumin, which comes in the turmeric family. Likewise, there are a lot of simple phenols, isoflavonoids. So there are a lot of common control points and diversity of related compounds that have emerged in different species to help with the same redox protection, stress management, and overall balance between the carbon assimilation and the redox function. Because this carbon assimilation redox function is the primary metabolic logic of the planet that we cannot run away from. So the more that's integrated into our food chain, it boils down to the solutions. So the take-home message from all these many years of work is that as the pentose phosphate pathway has to be regulated and balanced with the mitochondrial function, there may be a unique gate where this balance happens. And in our studies, we have looked at a proline-coupled regulation of the pentose phosphate pathway, which then regulates the anabolic counterbalance as plants face stress and reproductive needs. And so this balance between the carbon and redox protection is one of the central control point of plant metabolism and plant health. And we can couple this into our diet. So if this metabolic logic is coupled back into our diet, into our daily servings of fruits and vegetables that's recommended to be 7 to 9, 10 servings, then the same metabolism of plant benefits can be brought into our own diet. And so with that, we have applied this into type 2 diabetes, and which is a major burden or the primary burden of non-communicable chronic diseases, which then progresses towards cardiovascular disease, autoimmune disease, cancers, and all the other diseases, ultimately Alzheimer and cognitive failures, dementia. So this 90% of the burden is the adult onset diabetes, which is preventable by our dietary changes. Only 10% or so is where the pancreas is already not functional, and therefore you need lifelong insulin injections. But majority of the type 2 diabetes is type 2 diabetes. And this burden is only going to get increased and is projected to be about 600 million people in the world and taking a lot of our healthcare budgets. And the good news is plants have antidotes to this. So they have to work on the carbon metabolism themselves. In order to balance that, they have a lot of these glycemic control inhibitors that we can tap into our diet, whether it's glucose control, beta cell function, oxidative breakdown, blood pressure control, all of these different steps of the diabetes and its complication, we can get the antidote from plant foods. And we have applied this to gestational diabetes and child nutrition because this is very central, because we can set the child up for lifelong if maternal health and maternal burden of the same carbon burden is there in the womb. And therefore, gestational diabetes and consequence on child nutrition is an important facet of where we want to apply this. And another important fact is indigenous communities around the world have developed this very resilient, diverse food system. So what are the health benefits of them in terms of glycemic control or the consequence of glycemic control? So we have looked at the indigenous food system of our own North American communities. And we see this Three Sister Crop Foundation, which is colored corn, colored all the beans, and the pumpkin and squash foundation, has a lot of health benefits which are relevant for hyperglycemia, hypertension management, which are the early phases of the early onset of diabetes. And so with this foundation, we're also building this traditional Three Sister Gardens with high bioactive enriched diabetes countermeasure associated gardens, where we can target the health disparities within the American Indian community. And therefore, we're also looking at the child and maternal health nutrition and looking at their traditional food crops and seeing where are these bioactive enriched components are. And also from other immigrants who have come into America, how can we get a whole food, indigenous, traditional, or overall immigrant experience combination into these management of these chronic diseases, where early dietary interventions could be the most effective and cost-effective tool. So with this approach, we've also applied this with my collaborator, Dr. Ranila from Peru, into the Andean food system. And we are seeing how these traditional Andean similar Three Sister logic is also present in South America. And the colored corn, beans, and all the pumpkin and squashes also have similar role. And how could we bring this back into our dietary system? And therefore, can indigenous food diversity lead to gastronomy innovations? And the answer is yes. So can we bring food that support the microbiome and fight the bad effects of oxygen, high fiber grains, high fiber fruits and vegetables, good plant and animal protein balance, herbs and spices? And so we have already worked on some of these themes, preserving the traditional food approaches, and from which, how can we understand the bioactive components are there? So these gastronomic innovations not only are biochemically rigorous and rational, but also we preserve the indigenous traditions that had built these foundation of this system. And so with this logic, we are now trying to integrate this community-based approach into bringing our food diversity into modern sustainable production practices, preserving the traditional food systems, building in synergy based on the biochemistry, and optimizing it to the modern technologies we have learned to appreciate, microwave, what happens when we use these modern tools. So all these innovations are great, but how do we put it into a sort of a whole food approach, preserving our traditional practices? So with that, we have built a sort of a sustainable production, traditional food crop system, all the way from field greenhouse to the value chain. And we want to put this into every community on Earth, preserving their traditional food systems and preserving their local traditions, preserving their geographical indicators that gives them value. But bring in the rigorous biochemistry that is relevant. And one of the important is, how do we build the microbiome and the metabolic interface that not only allows plants to be resilient towards climate change, because they also facing the same oxygen burden as they meet the stress demands, but also how what plants produce for their stress countermeasures can be translated towards human protection. Okay, so our dietary solutions can come from the same logic. So resilience of plants towards climate resilience also shapes the human health and resilience. So this dual function approach and the relevance of the microbiome that affect both of them is one of the approaches we are trying to build towards climate resilience and human health. So this microbiome and redox biology platform is the design engine we want to take all the way from plant growth and analysis, science-based product design, optimal manufacturing, and then effectiveness for human health. So this discovery engine, we want to integrate to every community on the planet. And therefore, in summary, we need a systems, whole food, diverse approach to sort of counter many of the failures of our current food system, for which we need a sound metabolic logic based on redox biology and microbiome rationale. And using this microbiome ecology approach and the redox biology approach, we can apply to contemporary nutrition and food science solutions to combat diet-linked chronic diseases. And this systems biology-based approach is a problem and solution-oriented approach where plants are the foundation, and the carbon assimilation from the photons that the plants sort of use to split water, from which the carbon assimilation emerges, and then the basic nutrient platform is put together, are this foundation or the ecological foundation to meet global challenges, from which we can see all of the other challenges emerge. And so using this control point logic and systems approach, we can build more comprehensive approach. So that's all I had to present today. And I hope you found the conceptual logic relevant. And I thank you for your attention.
Thank you, Kalidas.
That's what it is.
Thank you very much for your interesting presentation. Unfortunately, there's no time for the question. We have to go on with the second speakers, David Colling. He's a professor of plant pathology at the Department of Plant and Environmental Science in the University of Copenhagen. He's currently president of the British Society of Plant Pathology, and he's here because he's one of the leading experts in the study of defense mechanism against pathogen in plant. And his research focuses mainly on understanding the fundamental aspect of fungus-plant interaction. And today he presents his work entitled Innovative Microbial Strategies, Enhanced Fitness and Nutraceutical Properties of Plants, the effect of the root, Endophytes, Serendipida, indica on specialized metabolism in tomato shoots. Please, Professor Colling.
Thank you very much for the kind introduction. So I'm going to focus my talk on a particular PhD project that I finished a couple of years ago, and the final publication has just come out actually quite recently. So the focus is on this endophytic root organism, Serendipita indica, And this actually has a link to the previous speaker, because this particular fungus has only been found in the wild once, as far as we know. And this was found on a bush in the desert in Western India. It is a model organism for root endophytes. It has been used in two rather different model plants, Brachypodium and Arabidopsis, and it's also been studied quite a lot in cereals, especially in barley, and also in tomato, which I will concentrate on today. It's in this order, Sabinicillales, in the phylum Basidiomycetes. It has an interesting infection pattern, which I'll illustrate on the next slide. It manipulates the host, both producing effector-like proteins, interferes with plant hormone homeostasis, and it also produces phytohormone analogs. And there's also increasing evidence that it's also using RNA interference as a way of manipulating the host. So first, to step back to endophytes. So what are endophytes? Well, these are basically organisms that live inside plants and all domains of life without necessarily causing disease. However, it is a gradient, so you can see that some of the organisms that can be endophytic in some circumstances can actually, under different physiological conditions, cause disease. And I'll come back to that towards the end of my talk. So these are microbes that live inside the plant without causing damage. In other words, they're commensal. And we find that some of these actually benefit the plant and help it grow. So it can be a mutualistic interaction. And one doesn't need to look for a more complicated explanation for this than the fact that if the plant is happy, then there's more habitat for the microorganism to live in. On the right, it's illustrated an endophyte growing out of a wheat grain onto water agar. So, and this is how you can study them, is use surface sterilized plant tissue, and if you're lucky, the endophytes will grow out onto the agar, where you can then characterize them. This talk is going to focus very much on terpenes, and terpenes are a very important group of specialized metabolites. And these include many of the smells and flavors that we have in our food and so on. And basically the biological role for these compounds is largely in defense and also in signaling for plants. And of course, as volatile, many of them volatile compounds, these can be detected for microbes, and they can react to these compounds to start for their own processes. So my former PhD student, Fanny Natala, did her PhD studying serendipity and its effects on tomatoes, and started by doing genome binding for gene clusters in tomato, which were of interest. Also did metabolomic studies to see which compounds were being produced. And it has done heterologous expression studies to identify functions for a couple of these genes encoding terpene synthesis, and working both with plant and microbial systems to do this. So serendipity, this is a root endophyte. It has a fairly narrow zone where it can infect, in this case an Arabidopsis root. It's taken from somebody else's work on Arabidopsis, where it actually invades some of the cells, and you can see sporulation inside the cells. It used to be called Piriforma spora, because the spores are supposed to be pear-shaped, although it's not particularly obvious. This one, perhaps you can call this pear-shaped. Get the right, across the right. I think I went 2 slides, yes. So basically, what Fanny did was using an experimental system with tomato seedlings. This was chosen so that she could study the volatile compounds. I won't come back to the volatile compounds very much, but isolating messenger RNA from the roots and the shoots of inoculated seedlings, harvested them, did RNA isolation, RNA-seq, and then mapped the transcripts of these compounds to tomato gene models, and a small percentage of these compounds also mapped, or the transcripts mapped to serendipity genome as well. And focused on the genes which mapped to the biosynthetic pathways for different specialized metabolites. And this was done in collaboration with Sean Johnson at Michigan State University. So it's a quite complicated slide, but basically it illustrates the different gene expression that was observed. So the left-hand panels is the gene expression seen in the shoots, and the right-hand panel seen in the roots. And I think the most remarkable thing about this is that there was a much greater induction of gene expression in the leaves than in the roots, but the fungus is only infecting the roots. It doesn't infect the leaves at all. So that shows a lot of signaling going on. As I point out, this is an enclosed system, so it may well be that it's the signaling to the shoot is a reaction from the production of volatile compounds. So several different biosynthetic pathways for specialized metabolites induced in the shoot, but we focused very much on the terpenes pathway, and these are some of the terpenes that are produced in tomato. And this is one of Fanny's pictures of the serendipity inside a tomato root cell. So she focused on the terpenes being produced and found that there were, the terpene genome of tomato had been characterized primarily by a Dutch group, and they were seeing about 30 different terpene synthase being induced. They function, some of them are functioning in the plastids and some of them function in the cytosol of the plant, producing different kinds of terpenes. So if you look in the induction of the genes, then in fact the majority of the genes were not particularly affected by serendipity, but the largest changes in gene expression were actually occurring with some terpene synthesis in the roots. But we can also see there is terpenes being produced in the leaves as well. So the main products are shown here. So you can see there's quite a diversity of different terpenes being produced in the system. One of the terpenes synthases that Fanny picked up in the transcriptome analysis had not been characterized at the time. And she used a heterologous expression system yeast to demonstrate that this was producing this particular terpene. But since this hadn't been characterized in tomato before, subsequently someone else found it and published first, so Fanny found it as well. And this is producing this particular compound. So the next thing she, sorry, I'm hopped. The next thing she did was looking at the transcripts that mapped to the serendipity genome, and she found one of the very first terpene synthases, a sesquiterpene synthase, in the fungal genome, and was able to show that characterize this and what it was doing, and was able to demonstrate that this was producing this compound called viridifloral. And viridifloral is actually quite interesting because it's a terpene that has an industrial value. It's a component of certain perfumes. So this shows the characterization demonstrating that it was this compound that's been produced rather than a related compound. This resulted, a study again done in collaboration with Michigan State, and they are better at patenting things. So there's now an American patent application for the use of this as a potential compound, production of this valuable compound, viridiflorol. So what is it doing for the fungus? Well, it would appear to be that the compound has an antimicrobial activity, and so this was an in vitro assay that was set up so that to look for this activity, and we can see that with this particular fungus, Colotrochicum truncatum, is inhibited by concentrations of viridiflorol above 100 micrograms per milliliter. And viridiflorol has to be solubilized in a methanol concentration in methanol to be able to be solubilized. And we can see here that the methanol does not affect the growth of this Colotrochicum fungus. So this would suggest that the primary role of this compound is to help serendipity to compete with other fungi that it's meeting in the environment. So if we're looking in the interactions between fungi in plants, then we can see there are very many different fungi present in the plant roots. And in another PhD project, we actually characterize these in tomato roots. Basically, our main biological interest is in trying to understand how microbial biological control agents can operate. And what we see is that the endophytes are often causing a mild activation of host defense mechanisms in the plant, but that when the pathogen comes along, then the plants are primed for their defenses, so they can react more rapidly to the plant defenses, to the pathogen, and actually prevent pathogen attack. And this particular, I've chosen here to take a leaf as a model, but it's the same kind of thing happening in roots. It's just the roots don't have stomata that the different fungi can invade through. So this was studied by Andrea Manzotti, who also did a PhD in our lab a couple of years ago. And he isolated a lot of different fungi from tomato roots. And his study was in relation to hormone balance. And I won't go into the details of this. But so what he saw was that quite a lot of the fungi that were present in the plants were potential pathogenic fungi. And when he isolated these and put them back individually into the plant, they could demonstrate these were actually pathogenic. So it's actually quite interesting here is that the microbial community in the roots was suppressing a lot of these pathogenic fungi until so that they weren't causing disease. But when you study them individually, you can see that they could cause disease on the plants. Okay, so to summarize my talk, or look at the prospects. So we see that microbes are stimulating specialized metabolism, and we think this is primarily to help them compete against other microorganisms which are present, and in some cases, of course, also to help infect plants. We see that there are many defense compounds being produced, and these are produced because they benefit the microorganism. But if you look in terms of nutrition, which is the main theme of the conference today, then we do see examples that some of the compounds that have been produced are actually quite healthy for this. And perhaps the best example of this is the stilbene resveratrol, which is a phenylpropanoid compound, which is in some systems preformed defense compound. In other systems, it's induced by microbes. So I think there's a big perspective to be able to use microorganisms as a way to stimulate the production of beneficial compounds in in plants, which can be beneficial to us. And if we look at endophytes, then we actually see this is quite an old paper, annual review article from 10 years ago, where you can see that the number of patents involving bioactive specialized metabolites produced from by fungi is increasing linearly over the years. I haven't tried to update this figure, but there's been a huge interest in the production of specialized metabolites by endophytic fungi. Okay, so to summarize, we see that Serendipita is a very interesting fungus. It can infect many different species of plant. It can help protect against pathogen attack. It's also interesting because it can confer abiotic stress tolerance, can induce resistance systemically against different pathogens. It has been so promising that it's actually marketed as a biological control agent in India, which is the country of origin. We're slightly wary about introducing microbes from one part of the world to the other. And then, of course, there's also the international agreements about if you found a particular organism in one country, then it belongs to that country. And Serendipita indica belongs to India. But related species actually seems to be promising to be able to do the same kind of thing. So the Serendipita vermifera is studied in Europe to do similar kinds of things. To acknowledge the project that was responsible for the 2 PhD projects I mentioned, I primarily presented Fanny's work as part of this talk, but I also mentioned Andrea Manzotti's work with tomato roots. And this was the early stage researchers of this EU project, Marie Curie early training network, when these are all the different PhD students. And then we are 3 supervisors who sneaked into this picture as well. Okay, so thank you for your attention, and I'm prepared to take any questions.
Thank you very much, David. You have time for a couple of questions. I just check in the web if there are some questions.
Yes, well, I could answer. Well, Mara Quigala has asked a question about looking at these antagonistic organisms for plant defense. Could we use them curatively? Yes, and I think that Sabrina will come back to that later this morning with the Fusarium system. But there are examples of some of these fungi that can be used curatively, but by far the largest number of examples we see is preventative biological control. Have we looked at the role of the microbiome community? Well, we have only studied, the 2 PhD projects we worked with in tomato were not looking at the role of the community, but we did look at the community in tomato with in relation to the hormone balance. So it's the different mutants and impaired in hormone production, and see how that affected the community, and then study some of the organisms that we found there. But there, our observation is that if we look inside the roots, then we normally don't see disease. But if we analyze the organisms that are present, then we can easily find plant pathogens. So our surmise here is that the endophytic microbiome community in balance is preventing the plants from getting sick.
Thank you very much. If there's no other question, we can go on with the presentation.
Okay, thank you very much.
Thank you.
Thank you, Daniela. Thank you to David. It's my pleasure to introduce Lilian Barros, that is a professor at the Centro de Investigación de Montaña, Instituto Politécnico de Brancasa. Actually, she is vice coordinator of the same center. Her research targets are mainly in the identification, separation, and recovery of functional molecules from different natural products. She has published more than 573 indexed papers in the food science and technology area, edited 4 books, written several book chapters, registered 13 patents, and is principal investigator of several national and international research projects. She also has reached the top position in the world ranking, such as highly cited Research Clarivate since 2016. The title of his presentation is Food Metabolites and the Nutritive Value of Crops. Please, Lilian, the floor is yours.
Thank you so much. First of all, I want to thank you for inviting me to show some of the work that we have been developing over the years. Today, I will speak a little bit about food ingredients and how they can be applied and how can they be able to reach these ingredients from nature and some of their applications that we have been doing over the years. So, all of you know that food processing and when we use natural ingredients, there are several questions that have to be answered and several steps that have to be performed. So when we use these natural ingredients, what we want to achieve is to have a higher food quality and safety. Also, by using natural ingredients, we can avoid the usage of artificial ingredients that are a main problem in the food industry, especially in the next following years, in which some of the preservatives will have an outcome of the by EFSA of their utilization in food products. So we have to find alternatives at this point in order to give us a higher quality in food and safety. So when we study natural ingredients, we also have to search and to study their sustainability in the food industry. And in order for us to apply these ingredients as an alternative. By using these ingredients, we are also developing new food products because consumers are reaching also for a green label, for a clean label in their products. They don't want to see the E-codes. So if we use different ingredients in the sense of as using them as ingredients of additives, without getting the E-code, we are acknowledging the development of new food products. And this gives a new trend in the food industry, which is the main outcome that the industry and the consumers want. To obtain these ingredients as also the food products at the final process, we are applying innovative technologies. And these innovative technologies will help us extend the shelf life. And this has a great convenience for both industry and also consumers, which we want a product that has a higher shelf life, but also has we can avoid a higher bio-waste production of the food. So as most of you know, there are several classes of food additives in which we have the preservatives class, the nutritional additives, the coloring agents, the flavoring agents, the texturizing agents, and the miscellaneous agents. Within my presentation, I will speak mostly about two main additives that we have been working at this moment, which are preservatives and coloring agents. A couple of years ago, we did a research and a review article in which we compared the different additives, and we compared the legislation between EFSA and FDA. And what we saw was that there is a contradictory between these two agencies. What we visualize is that some additives that are approved by EFSA, they're not approved by FDA, and vice versa. So when we look at this as consumers, we get a little bit of a conflict because we don't know why there's this difference between legislations. So our main focus of research until now has been mainly focusing on different natural additives that we can obtain from natural sources. They already exist. They are already allowed and permitted by EFSA in different classes. And between these different classes, there are certain natural additives that can be already applied. But what we are trying to find is their sustainability, trying to find different sources that can also give these compounds and can give these functionalities to food. So over the years, we have been studying different types of plants. Until now, we have studied more than 180 plant species, and we have a database with all the compounds and all the bioactivities that these natural matrices possess. And like I mentioned, I'm going to focus mainly on preservatives and on colorants. So when we want a great preservative, what should we find? We have to have an ingredient that has a high antioxidant capacity, but also it has to have a high antimicrobial capacity. So with this database, we can visualize and see which plants and which matrices could have these functionalities that can be used in the food industry. Also, we have been studying different mushroom species. Brescia, which is the city that I'm from, is really nearby a mountain, which is Montezinho, and we have a high diversity of mushrooms. Even this week, I went to the field to try to collect some new mushroom species to continue studying, to have a higher knowledge of different species that can be explored and then can be cultivated in order to have these bioactive molecules. So this is another source that we have been exploring in terms of natural ingredients. In order for us to have these natural ingredients, of course, we have to process and go through different phases. So like I mentioned, we have our plants, our mushrooms, our byproducts, because I'm not going to enter mostly the byproducts in this presentation, but for the last 3-4 years from now, we have been working with the food industry, trying to use their byproducts and try to process them and to have higher value to these bioresidues. Today, I'm going to mainly focus on plants and mushrooms. Afterwards, these natural materials have to be processed, in which we do an extraction. These extraction methodologies that we apply, normally we focus on conventional and innovative methodologies. We also apply different statistical models in order to have the highest yields of these molecules. After, of course, we do a chromatographic analysis, and also we study their bioactivities, and then we see what type of application we can give to these natural ingredients that we obtain. These are some examples of extraction methods that we normally compare and see if they're viable, and try to check their viability in terms of industry. Because you know that when we apply this in the lab, we have certain yields, but then when the industry wants to have a solution, we have to find them an easy method and economical extraction methodology that they can apply. So we try to see different methodology and compare them in terms of yields and in terms of economical costs. After incorporating our natural ingredient, of course, we have to go through different analyses in terms to see their quality, their safety, their toxicity, and also to see if in fact these functionalities as preservatives and colorants are actually going through to our products. So we go through different stages in the processing after the incorporation of the natural ingredient. So now I'm going to give you some case studies that we have been developing in our lab until now. This is an ingredient that we obtained from strawberry tree. Strawberry tree is highly found in our region. It's not very used. And it has a really high antioxidant and antimicrobial capacity because their main compounds are flavonols, derivatives of catechine and epicatechine. We studied the stability of this ingredient, but we also studied its incorporation, its stimulation in the incorporation of a food product, and saw that it was stable and could be applied. So this ingredient is now patent, and it could be applied in the food industry. Just to have a scheme of what we do when we receive the fruits of a strawberry tree in our lab, we normally try to extract, like I mentioned, using different methodologies. We have a response, and then we use the statistical models in order to give us the highest yield and the highest purity of the compound. And then, of course, we studied the stability and also their incorporation in the food product in order to see if it could be applied as a food ingredient. Here is another example in which we applied the previous ingredient from strawberry tree, which I mentioned was rich in catechins. And then we applied another ingredient from basil. And this ingredient from basil is rich in different types of compounds, more phenolic acids like rosmarinic and chicoronic acid. In this case, we are working with a bread industry from Bragança, and she wanted to have an alternative from loaf bread in order to avoid the use of synthetic additives. In this case, we studied these two, arbuto from rich in catechins and also basil, and we compared it from the synthetic ones that are used, which is potassium sorbate, but we used the natural one, which is ascorbic acid, to see the difference between the new natural ingredients that we obtained and also the ones that are already existent. What we found is that it had a similar capacity to ascorbic acid and to the artificial additive. And in terms of toxicity, it did not reveal any toxicity. And it had the shelf life was very similar to the ones that are already used. So they could be actually incorporated in this type of food product and to be used as a natural preservative. Another example here obtained from two different ingredients, which was one from fennel, rich more in flavonoids and also a phenolic acid, which is quercetin, and a chlorogenic acid. And then from another ingredient obtained from German chamomile, which is rich also in phenolic acid and lutein. In this case, we applied the ingredient in cottage cheese, and we compared it to a control in which we saw that these ingredients were also promising. In this case, we worked with a cheese company, not from our area, from Serra da Estrela, which is another mountain area in which these cheeses are very recognized in Portugal. Then we wanted to see the efficiency of these ingredients when we put a little bit of thermal temperature to our food product, in which we wanted to see if there was actually a type of degradation, and if we had to higher the quantity of the ingredient in order to have the preservative capacity. We had to do this. They had a slight degradation because we didn't encapsulate our ingredient. We try to avoid encapsulation if it's not necessary, because this will also higher the price of the ingredient for industry. So what we did, we hired a little bit more the concentration of our ingredient in this product. We compared it with the synthetic additive, and we visualized that it had a preservative capacity along the shelf life of the product. Another example is one obtained from lemon balm. In this case, the ingredient also was very promising, but we had a problem. It showed a little alteration in terms of organoleptic aspects, especially because of the color. To avoid this, we had to have to encapsulate this ingredient. But if the consumer is ready to approve a different type of product, it could be used in its free form. In this case, we also we incorporated it in a pastry product, a cupcake, and we also visualized that we had to hire a bit more the concentration of the ingredient in order for it to not have the degradation of the cooking process. But it also had a relevant preservative capacity in terms in comparisons with the control. These are the two most promising ingredients that we have found in our group. One is a coating material to avoid plastics. This idea won the European Innovative Prize in Brussels in 2019, in which we use all natural compounds in order to find a biofilm to produce this spray safe. What we do with the spray safe is to avoid, for example, plastic which is involved, for example, in ham in the supermarkets, in which we use this spray in order to put in this type of products. In order to have a higher preservation. What we visualize is with the spray, it preserves the ham during around 2 days in order to avoid the plastic, and then it's edible. So since it's edible, you don't have to wash it out and you can eat it. This ingredient has an international patent and is now being explored by a national enterprise in order to check the possibility of putting this spray safe in the market. Another ingredient that is already on the market and is already being used and developed a spin-off, which is an ingredient obtained from chestnut flowers as an alternative to sulfites in wine. So at this moment, this ingredient is being applied in several wines in Portugal, but not only in Portugal, in several countries in Europe. And it has been approved its usage in around 50 tons of hectoliters of wine at this moment. And now we're trying to approve this ingredient by EFSA. So here are two most promising natural preservatives that we have developed until now. Now I'm going to pass on to the natural colorants and their application in food products. Here we have an ingredient that we extracted from Glophrena globosa. This flowers, these petals are used for their anti-inflammatory activity. Actually, when we started studying these petals in the labs was with that intention, but then we saw that it had this great color, and then the compounds found in these petals are very similar to the ones found in beetroot. There are beta-cyanines. And since beetroot is already used in the food industry as a natural colorant, but it has certain disadvantages because of the taste and the odor that it gives sometimes to the food product, we started exploring Glophrena globosa as an alternative to beetroot. Here's more or less the scheme that we, like I mentioned in the preservatives, that we follow through. We do different extraction methodologies, but in this case, we had to do a separation of the petals and the cotton that is in between the petals, and we had to, for it to have a higher yield in the colorants that the Glophrena globosa has in its constitution. Just to know that the compounds found in these petals have a higher stability in comparison to beetroot, because the beta-cyanines that are found in Glophrena globosa are acylated. So since they're acylated, they have a higher stability when incorporated in the food products compared to the ones found in beetroot that are not acylated. Then afterwards, we incorporated the beetroot, the ingredient found from Glophrena globosa, and also a control ingredient that we bought, a natural ingredient from the an enterprise. And we worked with a national company in order to produce red velvet. And we saw that effectively this ingredient is comparable to beetroot, but there is a bit of a problem with the Gomphrena globosa. It's the sustainability, because we don't have high quantities. We would have to have fields and fields of production of Gomphrena globosa in order for it to be sustainable, in order to substitute beetroot. But it's a possibility online that researchers and that companies have to see if it's viable, its application as a natural colorant. We also applied this ingredient also in different products. Here I have ice cream, but we also applied it to different pastry products. Here's another example, Roselle, in which we also extracted it and it had a high colorant capacity. We applied it in different food products, also with the baking industry, and it also showed it to be very promising. Here's another example. In this case, we use figs, the peels. In this case, we use the bioresidue, because all of us know that from the production of fruits, 20% of production are residues. And this company spoke with us, the fig production company spoke with us and asked what alternatives they could have with their residues in order to have a higher add value product. And we extracted cyanidine from figs, and we applied it in a donut glycine and gave them that. It doesn't have to only be in a glycine, it could be in different incorporations, but here we wanted to show the company that they could extract cyanidines from the peel and have this colorant capacity. In terms of, I've already spoke with two different types of compounds, beta-cyanines, anthocyanines, and I'm going to give an example of carotenoids in which we extracted curcumine and also the carotenoids from paprika, and also the beta-carotene and lycopene from tomato. In this case, we the bakery company Terpenes that we worked with wanted to do a cream, a pastry cream, and they wanted to avoid the synthetic ingredients. So we gave them this alternative in which we joined these three ingredients together in order to have the color that they effectively wanted in their food product. Now going on to another type of ingredients, which are the natural bioactive compounds. I know this is more a plant-based conference, but I had to show this example in terms of mushrooms, in which we isolated a sterol, in this case a mycosterol, which is ergostrol, which has an epiclosteremic effect. This already exists because we have the phytosterols, but the phytosterols are really already very explored, and also for their extraction, the extraction process is not very green, which in this case of the mushrooms, we are using the residues from the mushroom production from an enterprise here in Brescia, also, which is called Morgaricus, and we applied a greener extraction methodology in order to extract these mycosterols, and we apply them to a yogurt in order for them to have these effects. Here's again another scheme of what we do in terms of their extraction, in terms of extraction methodologies that are applied. And then afterwards, its application in yogurt. We study the stability along the shelf life. And also when we study the stability, we apply always the processes that I talked to you in the beginning of this talk. So, another product that we can obtain from mushrooms, we can obtain the ergostrol for its epiclosteremic effect. But if we give them a UV light, the ergostrol is the precursor of ergocalciferol, which is vitamin D2. So it can also be used as a regulation of calcium and potassium. So this is another ingredient that we can obtain from the residues of Agaricus bisporus. Both these ingredients have been explored. I already mentioned that we apply them to yogurts, but we also apply them to cheese products, to cured cheese, to cottage cheese, to fresh cheese. But we also applied the vitamin D extract to bread products in order to fortify it and to have the necessary doses of vitamin D in one bread for it to cover the normal ingestion of this vitamin. Also, regarding the Ergostrol, when we apply them in these cheese products, what we notice is that there was a decrease in the absorption of cholesterol by 30%. So while you may ask me, why cheese? Because cheese has cholesterol. So this can give us an advantage because we're ingesting a product that already has cholesterol, but we are having an ingredient that can lower the cholesterol while we're ingesting this type of food product. What we notice is that the ingredient only has a stability for 9 months. All of you know that a cured cheese could have a validation date up to 12 months or more. But in this case, if we incorporate the Ergostrol in the cheese product, it won't have a higher duration than 9 months because the ingredient will start degrading. In terms of natural bioactive compounds, if we wanted to have these capacity, we have to encapsulate. That's the problem when we work with phenolic compounds because they're highly metabolized. So here I give you some examples of encapsulations that we have been performing before incorporating these natural bioactive compounds in the food product. Here is an example where we extracted an ingredient from wild strawberry, from Fragaria Brescia, in which it's really rich in tannins and also flavonoids, and then we encapsulate it and we incorporate it in gelatines. Of course, when we encapsulate it, it has a higher bioactivity when we study bioaccessibility assays, because it will be protected throughout the digestion process. Here's another example in which we also encapsulated this ingredient, also rich in tannins, especially elagitanins, and we incorporated this ingredient in yogurt. Of course, it was more bioaccessibility. It had a higher bioaccessibility when incorporated than when not. So another link software that we developed was this one that you're seeing here. The link is www.decode.ipb.pt, and where you can search different additives, the ones that we have already explored, the ones that are already on the market. You have all the information, its toxicity, its dosages. This website is already being used by different companies in Portugal, especially a really big one here in our country, which is Sonai, that has been using this software in order to see what different alternatives they have for additives. Of course, none of this work, you've been seeing many pictures of different students and different researchers of my group throughout the presentation. None of this could be possible without the work of a really big group, which we have been working together for several years now. I also want to just let it out here that anybody has interesting works in the field of food and function, they are also invited to submit their works to our journal. So overall, I want to thank you all for listening to me, for listening to all this work that we have been developing in terms of natural ingredients, and thank you for the invitation for today.
Thank you, Lilian, for your important, interesting contribution. We are in time, so if we have some question from the floor, please.
I have a couple of questions. Thank you very much, Lilia. Very, very interesting presentation. The first, just a curiosity. How this extract you added to the food can interfere with the flavor of the food? And the second question is more general. From a regulatory point of view, how these ingredients added to the food are framed?
Okay, So for the organoleptic part of the question is that, like I mentioned, for example, the extract used from Melissa, it had a problem. It gave a little bit of color. But in terms of flavor, it didn't affect the flavor of the compound, because even though we use, when I say that we use higher quantities, the quantities are low. Okay, they're not very high in order to give the flavor to the food product. But to overcome sometimes these problems, because we do find these problems, especially when we use extracts with high flavor. For example, if we used Oregon, or if we used thymus, for example, tomillo, I don't know how, I can't remember now in English the word, but I think it's thymus. It has natural flavor. Okay, so in this case, we have to encapsulate. There's no other way to overcome the organoleptic properties of these ingredients. But the ones that I've been showing in the presentations are those that don't give flavor to the final product in the doses that we have been applying to the product. But of course, we have seen these cases of flavoring in different ingredients that we have been exploring, but in that case, they're not very promising, because in fact, it has to go through an encapsulation process. And like I mentioned, the industry wants to normally avoid this encapsulation. In terms of regulations, this is quite, it's a really good question. And of course, like I mentioned, for the ingredient obtained from chestnut flour, we are going through this process at this moment. It's approved at this moment in the winery sector in different parts of Europe to be applied. But now it has to be applied. If we want to go to higher quantities of wine, it has to be approved by EFSA. And this process is very complicated, as all of you know. And we have to do certain assays that we have already been applying and that we haven't been performing at this moment. But it's a process that will take a couple of years for it to be approved. Then we have a different part, because the industry, in this case, if it goes through EFSA, it will have an E-code, okay? It will have an E-code. But the industries, what they speak with me about, what we have been speaking, is that they don't want the E-code. They want a clean label. And if we go through EFSA, we will have to have an E-code. So we have to explore these ingredients as a different basis in order for them to be applied in the food companies, in order for them to not have an E-code. So that's what I've been exploring now with our national nations in terms of legislation, which is ASAI. Probably all of you have, and we're trying to see how we can approve these ingredients and not have the E-code. So it's a process that we are working with, but if we don't start working with this process, nothing can be done. And in a couple of years from now, we won't have alternatives in terms of applications. This doesn't apply to the colorants, okay? Because when we speak about colorants, we speak about groups, okay? About anthocyanins, for example, and they're already allowed. So if they're already allowed, and they're already allowed in certain quantities, as also beta-cyanins. So when we're using ingredients that are already rich in these types of groups, these ingredients can be applied. Okay, so this is more in terms of preservatives in which the compounds are not completely approved, but we are working in this process.
Thank you, Lilian. We have no time for another question, so we can go to the next speaker, that is Firat Gunder, that is a professor at the Department of Bioengineering at Imperial College London. Firat and his team work in the interface of material science, electronic chemistry, and biology. They focus on the development of new materials and the fabrication of low-cost sensors and actuators with the aim of transforming the device developed into fully functional portable system for use in healthcare, agriculture, and food science. In addition to his scholarly activities, Firat has also co-founded multiple startups to translate his research to the real world problems. The title of his presentation is Sustainable Sensor for Agriculture and Food Monitor. Please, Firat, the floor is yours.
Thank you very much. Can you see my screen?
Yes.
Perfect.
Thank you.
Okay. Good morning, everyone, or wherever you are. Good evening, if you're further east. So I changed the title of my talk slightly. But anyways, I will be talking about more or less the same areas. So it's a great honor to be here. In fact, actually, I collaborated with the University of Brescia before. It was a fantastic opportunity to come back and then share some of our work. And in fact, one of the works I will share is it was carried out in collaboration with the University of Brescia, and I co-supervised a PhD student there. And also, it's interesting because, for example, there was a speaker from the University of North Dakota, and in fact, I was a high school student for a year in North Dakota, which is ironic because it's a very small place in many ways. Okay, so I would like to start with this, very familiar picture, which is in some ways quite beautiful. When we walk into a supermarket, there are shelves full of food, and then it really greatly simplified our lives to have access to such resources. Most of the time, just a few minute walks away, and it really created a system for us where we no longer need to spend a lot of effort to have access to high quality, safe foods. However, this beautiful picture has a dark side, which is that it gives us the wrong impression that everything is solved and it's all good and clear and beautiful, and we don't really have any problems about the way we produce food and we consume food. So in fact, one of the biggest issues regarding food is the waste. So a third of all food we produce globally actually gets lost. We never consume it. We just essentially throw it away. It ends up in the bin one way or another, or it's just never harvested or delivered to the individuals. So, for example, if we look at the UK, which is a relatively small country, 7 million tons of food is wasted annually by the households alone. And the interesting thing is that 60% of these foods are completely good to consume. There's nothing wrong with them. They just get tossed in the bin. And obviously this has a huge price tag, which is approximately in the order of 12.5 billion pounds per year. And by the way, these numbers are a couple of years old. And with the sudden increases in the price of food and cost of food, obviously it's the numbers are expected to be a lot larger in the coming years. And also there is there's another element too. So there's the cost, obviously, which is pretty bad, but there's also the environmental element of food waste, because food production is very energy intensive. Food waste alone is it's essentially equivalent. It produces equivalent of 10% of all greenhouse gas emissions in the world. So if food waste were to be a country, it would be the third biggest emitter of carbon dioxide after the emissions by the United States and China. So it really is an incredible problem. So when we started looking into this, one of the things became obvious, which is that, when we buy a food product, when we buy a pack of meat from a supermarket, it always comes with a date attached to it. Sometimes it's the use by date, sometimes it's the expiry date, sometimes it's the best by date. It's confusing, and it actually, this date, it does not actually tell us much about the biochemical the properties of the food itself. It's just a conservative estimate of approximately how long the product will be safe to eat. It's conservative because we don't want people getting sick from it. However, with this conservatism, we really end up tossing a lot of food in the bin, although it's completely okay to eat. In fact, like I've run some experiments myself. I stored yogurt at room temperature for 3 weeks, and multiple weeks actually passed its use by date, and it was still okay to eat. I didn't get sick. Of course, I don't recommend that people do this, but in fact, some of these dates are very, very conservative. So, and the use by date is static. It doesn't change if you leave your food on the kitchen counter, for a day, or you store it in the fridge, and clearly for various products, the storage temperature will be quite important, and other, or the humidity and so on, depending on what the food product is. So what people have been trying to do is to come up with intelligent packaging solutions. So these labels would essentially be attached to a food package, and it would change color, either change color based on temperature, so what temperatures the product has been exposed to, or other properties as well. And the most popular way of doing this has been to do it colorimetrically. For example, there is a product here called Fresh Meter, which is just a temperature indicator. However, although the idea is fantastic, and you can just look at the color and you can tell what's going on, you don't see these technologies out there. And a lot of people have attempted doing this colorimetrically, but they all failed, because it turns out that it's not really only a technology problem, although it is somewhat important to develop lower cost technologies. But it turns out it's just one of the elements of actually getting these technologies out there and bring them to the market. So when we talk to food producers and retailers, they told us that such technologies actually lead to more food waste, not less, because even the slightest discoloration leads people not to buy that product, because everybody wants always to buy the freshest product. So in this case, for example, giving too much information to the consumer, for example, it can lead to more food waste. Although there's absolutely nothing wrong with this product to eat. It's just that there's just the slightest discoloration. That's it in the tag. And oftentimes also these tags, they change color from red to green and so on. And given the fact that a lot of men are colorblind, it's actually it's not actually that useful because they wouldn't be able to tell the colors anyway. So when we started thinking about this, we realized that, well, whatever the technology is, it needs to be low cost and ideally disposable, because most packaging is disposable. It should not be visual. Ideally, it should be electrical, or even compatible with, for example, some of these technologies that are becoming common. For example, near field communication, NFC, that's used in wireless payment cards for contactless payment. And then it should obviously also be eco-friendly. So we don't want to basically solve a problem somewhere and then create a problem elsewhere, perhaps even a bigger problem. So, and considering all these factors, we have developed a technology which is based on paper, and it really costs, we call them near zero cost sensors, because they are extremely inexpensive, which I will go over in a minute. And this work was published a few years ago. It was led by my former PhD student, Gianandrin Barandun, and we got lots of help from our Italian partners in Brescia, and the student that was actually leading this work that I was co-supervising, it was Matteo Soprani. So he's actually still in Brescia. Yeah, and this work turned out to be receive a lot of attention. So, and the sensors kind of look like this. They consist of nothing but paper and some printed carbon electrodes. There are absolutely no other additives to functionalize, etc. And we can print them in large quantities. Hence, they are very, very low cost at the end. So, and the way these sensors work is that these are electrical sensors. They don't change color. So, if there is a spoiling food, it emits gases, and the sensor detects these gases, and it basically the impedance or the electrical resistance, let's say, of the sensor changes in linearly with the concentration of gases that are in the immediate environment. So basically, to detect the concentration of a spoilage gas, all we need to do is measure the impedance or the electrical resistance of the sensor itself. And the main idea of this whole technology is that paper is always wet. So, and the way I came about this problem, or this, I guess, solution, is several years ago, I was going through an old papermaking book, and I came across this plot that I show, which I basically copied and pasted from the PDF version of that book. And what it shows is that the moisture content of paper is it's actually it's highly dependent on relative humidity, which makes sense. But what's surprising is that paper is very hygroscopic, and it retains lots of water within it at relatively low relative humidity. For example, at 50% relative humidity, paper contains 5% water by weight, and that's a lot. That's bulk water. So, which means that although cellulose paper looks and feels dry, it's always wet. So that means that we can do wet chemistry with it. So that's kind of the basic idea. So we have a seemingly dry substrate that's always wet. And the way kind of these sensors work is that I'm not going to go into the details too much, but if we have a water soluble gas that's in the environment, it just goes and then interacts with this layer of water that's sitting within the paper substrate. So sitting on the cellulose fibers essentially. And then if this gas is let's say an alkaline gas, although it's not a very correct term, but it's a gas that can go and dissociate into ions and heavily dissolve in this layer of water. Then by measuring the electrical properties of this layer of water, meaning we just really measure the electrical properties of paper in this case, then we can probe the concentration and presence of various gases. And it turns out the sensitivity of these sensors are highly correlated with the Henry's law constant, which basically is a metric that tells us about the that's a relationship about the amount of a gas that's present, or concentration of a gas that's present in gas phase, and also that's dissolved in the solution that's in the same environment. So, and I think this was the really experiment that told us that this is an incredible technology. Although it costs nothing to produce these sensors, with just a piece of paper, we could sense very small amounts of, for example, ammonia, which is a highly water-soluble gas, but it's a very important gas. It has many applications. But we were able to go down to ppb, so parts per billion ranges in terms of sensing this analyte, for example, with just a piece of paper, and there's absolutely nothing else. And when I saw this plot, I knew that we were really onto something, because this kind of performance, we struggle to get even very high-end sensors. And here we have a sensor that's printed and really costs pretty much nothing. It's very inexpensive. So, and these sensors are it turns out they are the paper sensors are sensitive to a range of gases. But again, as I was describing before, the primary thing that defines the sensitivity in this case is the water solubility. So it's the Henry's law constant. So if a gas is highly water soluble, then the sensor is most sensitive to it. And so when we ran, for example, experiments with ammonia, trimethylamine, carbon dioxide, and other gases, we see that there is really a very nice correlation between the Henry's law constant and the sensitivity of the sensor itself. For example, carbon dioxide produces a signal, but only upwards of a couple of %. Ammonia produces a strong signal, even for a few ppm. Whereas, for example, carbon monoxide and hydrogen sulfide, they don't produce a strong signal or a signal whatsoever, actually, in the ranges that we tested, up to some sub percentage concentrations. So then we applied this technology for the detection of and monitoring of food spoilage. And one of the interesting things is that, as I showed you before, so the amount of water in paper, it actually changes with relative humidity. But monitoring food spoilage is really the perfect application for these technologies. It's because for this technology, it's because the relative humidity in a fresh pack of food is fixed at 100%. So that means that we don't need to worry about that change and that parameter. So, and furthermore, most existing low-cost sensors, they actually struggle at high humidities. But we have a technology that actually works best at high humidities. So that's why it's particularly suitable for food and biological applications, where everything is pretty much happening in water. So, which is the basis of our life, really. And you don't need to worry about this plot so much, but what we really tried to show is that we are able to detect food spoilage at relevant ranges, for example, in terms of microbial contamination. So, for example, the bands here, they show the microbial cutoff ranges for food products. We ran microbial cultures in parallel to the experiments, and these sensors were able to detect spoilage sensitively enough to be able to give indication or equivalent, let's say, results to microbial cultures. And I'm happy to, of course, go over these in more detail if there are more questions. But one of the things that we also tried to do is, okay, we have a sensor that seems to work well, but what do we do next? So we said if we really want to increase the adoption of these technologies, we need to make them very easy to use. So we created a setup where we took an existing near-field communication tag. Again, this is the same technology that's used in wireless contactless payment cards. And then take one of these tags and then put the sensor across the chip that's already on this tag. And these tags are very inexpensive. I mean, they can be bought as low as 5-6 cents. And then, so when the concentration of a spoilage gas increases and passes a certain threshold, then the majority of the power goes through the sensor and no longer the chip. So the power is obviously wirelessly transmitted in this case. And then it turns the sensor into an on-off type spoilage detection system. But anyway, so Reuters came to our lab and they made a small segment about this. So I want to share this with you. Okay. How can we do this? let's see. Okay, here we go.
When there's so much of it on show, the temptation to buy too much food is very real. And once it's gone past its sell-by date, it often ends up in one of these, even though it's actually safe to eat. Scientists in London think they have the answer to stop this happening. Using your smartphone, you'll soon be able to find out if the food in your fridge has gone off based on the gases coming out of it.
I would never have thought of that we would go this far. But now that everything works, it actually looks like there's going to be a great impact. And we can bring it from the lab, where it works really well, to the industry and have retailers and customers use our technology.
The paper-based electrical gas sensors have been developed here at Imperial College London. They're made by printing carbon electrodes onto cellulose paper. They contain what are called near-field communication tags and microchips. That means information on the gases it's detecting can be read by your mobile phone.
The beauty of it, obviously, is the simplicity. We use paper, everyday paper, and just print an ink on it, and then we can measure gases.
The sensors cost about 2 cents each to make, and the developers say supermarkets could be using them on food packaging within 3 years.
All right, so yeah, for more information, please refer to the paper. And I guess the interesting thing is that when Reuters came, they actually interviewed me quite a bit as well, but I'm barely in this video. Yeah, maybe I'm just not good looking enough for TV. But anyway, so yeah, most of the talking was done by my PhD student back then, Jandrin. So just moving ahead, mindful of time. So the next thing I wanted to talk about was, so we try to develop a technology for reducing food waste, but I wanted to also take a step back and go to the beginnings of food and try to investigate really what's happening there in terms of food production. So, you know, if we go back, let's say 50 years, and we go to a field, we see that nothing really has changed in terms of the way we do agriculture. It's just that the only thing that's changed significantly and very visibly is the size of the machines. And then what we actually have been doing, so we've been increasing food production year on year, but one of the methods that we have applied is just that we add more nutrients. So we just do agriculture more intensively instead of using less intensive methods that you know reduce the productivity of the food system. However, this intensification obviously comes at a cost. We degrade the soils, etc. And in fact, for example, if you look at the fertilizer use in the last 50 years, it has increased 600% decade on decade. And it also shows that really we're not solving the problem in a sustainable fashion. All we're doing is just, you know, we're adding more things into the soil that would lead to higher production. However, that's at the cost of the soil itself and sometimes the environment. So I got interested in nitrogen fertilizers because they are the most used ingredient in agriculture, especially for crops that don't fix nitrogen themselves. I mean, and when I looked into the nitrogen fertilizers, I came across an interesting study which showed that in fact the synthetic nitrogen fertilizer was probably one of the biggest drivers of population growth. And the authors estimated that if the nitrogen fertilizer was never invented, the world population would be about half now. This exponential growth in population was really primarily supported by the use of low-cost synthetic fertilizers. But actually, if we investigate the nitrogen fertilizer, we realize that the application of this very important tool in agriculture is not really very well understood. So the growers often only follow guidelines. They don't really make any measurement of the soil of nitrogen or the concentration of nitrogen in soil. So oftentimes, it's the government that tells them, okay, you need to put X kilogram of ammonium nitrate or urea or whatever per hectare, and that's what the growers do. And it's so there's so much confusion about the use of nitrogen fertilizers is that this confusion leads to, or lack of information, leads to a lot of variation across the globe in terms of how it's used. For example, 8 times more nitrogen fertilizer is used per hectare in China than Australia. And it's just because of the practices that they have adopted. So excessive use of this fertilizer is incredibly common because farmers, they don't want to miss out on the yields because of lower concentrations in the soil. And because people are just following guidelines, lab testing is very uncommon for knowing how much fertilizer exists in soil, and then to top it up, let's say, synthetically and so on. And this excessive use of nitrogen fertilizer is really, it's degrading the soil, and then it's leading to environmental problems, huge environmental problems. Like, for example, in the UK, more than 50% of all land is a nitrate vulnerable zone because of farming. So when excess nitrogen ends up in bodies of water, it leads to algal blooms, such as the one here, which leads to hypoxic conditions and death of various sea life. So it's really not good for our ecosystems. So we have developed a technology for determining soil nitrogen with low-cost sensors and machine learning. And this work was led by my former PhD student, Max Grell, and we published it not long ago, actually. It was towards the end of 2021. So really the problem we try to tackle is how much fertilizer do we need to add to support plant growth? And we don't really want to add more or less, just try to add the right amount. So for this, to answer that question, there are really 2 other questions that we need to answer. Is what are the soil nitrogen levels now? And what will they be in the future? If we can answer these 2 questions, then we should be able to decide how much fertilizer to add to the soil. For example, if we look at the nitrogen fertilizer, whatever we add, it often ends up in 2 forms, in ammonium and nitrate, both of which are taken up by plants. However, this is an incredibly complex system. For example, if we use urea fertilizer, the conversion into conversion into ammonium, and then further nitrification into nitrate. These are very complex processes, and therefore the ways people have looked into this for a long time. Can we actually try to estimate using various mathematical approaches what the levels will be in the future and so on, or now? It's been incredibly difficult because most people have tried to use deterministic mathematical approaches and not statistical. So the way we approach this problem is we said, okay, we have an ammonium soil sensor, which we invented in the lab. I'm going to talk about it in a minute. So can we take the ammonium measurements and then also make a couple of other simple measurements in the field, such as the soil pH and electrical conductivity? And then we also take some of the readily accessible information, such as temperature, soil dryness, rainfall, time since fertilization, etc. And then can we throw this into a machine learning model of soil? And then from this predict the levels of nitrate, because we could already measure soil ammonium, and then predict the instantaneous levels of soil nitrate, and also future levels of soil nitrate and ammonium. So basically nitrogen levels in the future. So this is the kind of technology we try to implement. So again, the first critical thing that we wanted to do, or we tried to do, is try to make a low-cost ammonium sensor, soil ammonium sensor. And this is based more or less on the same technology that I presented earlier for food monitoring. It's just that this time we functionalize the sensors with some sulfuric acid. And the paper sensor basically detects the soil ammonium, but in the form of gas. And the change in conductivity of the soil, the sensor gives us an indication of the soil concentration for ammonium. And our sensors basically kind of look like this. We again print them, and these colorful bits are wax barriers to contain the hydrogen, the sulfuric acid. I'm running out of time, so I'm going to have to speed up. And the sensors themselves, they work. They produce an analytical signal that's actually within the desired ranges that would be useful. So I'm going to skip ahead. So then, using the inputs, so ammonium, electrical conductivity, and pH, we trained machine learning models so that we can predict the nitrate levels now and nitrate and ammonium levels into the future. So to basically train the machine learning model, we had to collect a lot of data. I'm not going to go over this. This is training data for the model, and then we built the model. But yeah, so the most important slide here is probably this and the next slide, that we were able to measure, we were able to predict the soil nitrate concentrations with an R² of 70 in comparison to the external lab measurements. And really the significance of this work is that without using a physical sensor, a software-based sensing approach, we were able to extract critical information from the whole system, so we can really predict the nitrate levels. And it's depending on the quality of the inputs, we were able to go up to an R² of 0.86. So for the software-based sensing approach, and not a hardware-based sensing approach. So this was for the instantaneous levels. And again, we designed we implemented also a model that can give us predictions for the future levels. Once again, I'm going to skip ahead, but this approach also seems to work quite well. So from measurements for measurements performed on day one for nitrate and ammonium, our model predicts up to 12 days the levels of ammonium and nitrate in soil. Okay, I mean, I have a video, but I'm not going to show you this because I'm out of time. So I'd like to thank my funders and obviously my group who have done all this amazing work. And we're also a very entrepreneurial lab. The food sensor that I showed in the first part of my talk is being currently translated and commercialized by Black Bear, which was co-founded by my former PhD students, including Max and Jandrin. And also, we have various postdoc positions available in my lab. So if you're interested, please do get in touch. With that, thank you very much. And sorry, I ran out of time.
Thank you, Firat, for your very interesting contribution. A lot of questions I have, but we have no time, and I hope to have the possibility for a speech with you to do some other question and some other consideration. Now it's time break, so we do not have time for questions, or if exists a very, very short question from the floor. You have?
Yes, I have. Just, I said I'm a pharmacologist, not an engineer. So probably my question could be a bit odd. But I'm wondering about the versatility of your sensor. Means that could be also built up for other species, not only for nitrogen species.
So do you mean, so?
Yes.
Okay, for soil sensing, we have only investigated at this stage nitrogen. And the reason is because it's the most used soil input. So people have, for example, also asked us to look into phosphates and so on, but these are usually not added all the time. And so our sensing platform, it's a general platform. We could obviously, you know, also make a model that includes, for example, phosphate levels, etc., and try to predict those. Our current sensing, our current sensor cannot sense phosphates, for example. So we would have to probably develop slightly different technologies, hardware-based technologies, to help with that. But our general approach, the software-based approach, should work for most nutrients.
Thank you, Firet, again. Now it's break time, so please come back at 11:45 that we have the second session. Thank you to all participants. Thank you to all the speakers. See you later. Okay, it's time to begin. Yes, it's time to continue the second section. So the next speaker is Daniela Bulgari, that is a professor at the University of Brescia. He is great and wide research activities, and two of her research activity are the study of the microbiome associated with apple and grape wine plants of different health status. The second in importance, neglecting the other one, are a study of secondary metabolites in filamentous fungi with particular attention to the regulation and functional analysis of glioxin. Some of the results obtained from this activity have been published in 26 peer-reviewed international scientific journals, 2 book chapters, and 30 contributions to national and international conferences. The title of her presentation is Microbiome Enriched Compost, Potential for Plant Resilience and Nutraceutical Application. Please, Daniela.
Good morning to everybody. And first of all, I would like to thank the organizer for giving me the opportunity to present our results on the potential application of microbial enriched compost in the induction of plant resilience and in the enhancement of nutraceutical value of crops. As in human, plants teem with microbes. And the microbial community is mainly composed by bacteria, fungi, actinomycetes, mycorrhizal fungi, and its complexity and dynamic, but also its functionality, is influenced by different parameters such as plant genotype, soil type, land management, but also this community is shaped by the presence of plant pathogens. The study of microbial composition and microbial dynamic is very important, but the more interesting thing is the crosstalk, the molecular dialogue that is that there is between the microbial community and their hosts. This crosstalk, this functional bond, lead to an enhancement of plant productivity, plant resilience, that means plant resistant to abiotic stresses such as drought, salinity, or the plant resistance to biotic stresses such as plant pathogens. Recently, it was also demonstrated a link between the microbial community, the functional microbial community, and the nutritional value of crops, which is the most studied beneficial microbes in agricultural application, the genus Trichoderma. Trichoderma is a fungal genus that has a very long history because it shows a lot of interesting features, such as the production of secondary metabolites or enzymes that are involved to the plant health status or involved in the newly or very interesting commercialized or marketed biofungicides or biostimulants. Recently, Trichoderma treatment were also used for study the involvement of this genus in the induction of the production of nutraceutical aspects of the plant. And in detail, as shown in this figure, it shows that Trichoderma treatment are able to increase the amount of carotenoids, polyphenol, glucosinolate in lettuce. Until now, the scientific community focused its attention, put a lot of efforts in studying the microbial application in field condition, in laboratory condition. But all these positive aspects of microbial community and plant interaction lead to the enhancement of plant productivity are less effective when we move from the laboratory to the field. Why this reduction in the positive effect of microbial enriched product? The reason maybe is in the application. Usually the concept is based on one microbe based application, while it's more interesting the use of synthetic microbial community. SYNCOM are small consortia of microorganisms designed to mimic this functional structure microbiome in natural condition. Which are the advantages of using a microbial consortium over one microbe application? The first is the increase in community stability. These SYNCOM are more stable, and then we have an advantage to colonize an environment that is rich in other microbes. And as we heard before, there is an equilibrium inside the field, and we have to maintain this equilibrium and give to the plant an advanced we have to give to the plant resilience to plant pathogen and also climate changes. Another interesting thing to this microbial SYNCOM, this synthetic microbial community, is the reduction in the complexity. This reduction in complexity could be achieved by the use of a beneficial microbe such as Trichoderma. What is another important aspect to increase the efficacy of this microbial-based product in their field? The vitality of this consortium, of this consortia, which is the aim of our work. The final aim of our project is to test the efficacy of microbial enriched compost in the induction of plant resilience, but also to increase the nutritional value of the crops. To achieve this ambitious goal, there is a necessary first step, that is the development of a protocol to obtain a stable microbial enriched compost to be spread into the field. This is the rationale of our work. We combine different kind of waste, the digestate, that is the byproduct of the biomethane production, wood sawdust, and agro-food waste. We use this waste as single ingredients or in combination as solid state fermentation substrate. These substrates are inoculated with Trichoderma, and after the growth of these microorganisms, we tested the amount of the beneficial microbe growth on these substrates, and then we tested its interaction with the plant by using the germination index. As you can see in the picture, we tested 4 different Trichoderma species on digestate and on digestate in different combination. And as you can see, we achieved the growth, significant, very high growth of Trichoderma only in the digestate amended with sawdust and food waste. We quantified the fungal biomass using real-time protocol ad hoc setup. And as you can see in the table, we found that this Trichoderma species grows in a different amount on this waste substrate, based substrate. And interestingly, we found that Trichoderma reesei grows in a higher amount, while Trichoderma afroartianum grows less than in comparison to the other. Then we tested the ability of the substrate and the crude extract to enhance the seed germination and the root elongation of different species, plant species. We tested first the activity of this solid state fermentation substrate ATTs, or diluted in different amount of a universal soil. And the germination index represents the ability of these substrates to increase the seed germination and root elongation assay when when the score of germination index is up than 100%. Above 60%, this index showed a phytotoxicity. We tested also this ability in the crude extract, how we obtain the crude extract. Crude extract is an aqueous extract obtained adding water to the solid state fermentation substrate. And also we diluted this into water. As you can see in the graph, in both cases, the germination index increased in a dose-dependent manner. It means that low doses of both solid state fermentation substrate and crude extract give the most positive effects. Interestingly, Trichoderma afroartianum at lower doses showed a significantly enhancement of seed germination and growth elongation. We used the same assay with the same germination index to test the ability of the substrates and the crude extract to enhance the germination of tomato plants. And as you can see, we found a strong enhancement of tomato seeds germination and the root elongation when we use the crude extract, also at the higher doses. But as I said before, the most interesting thing is not to stay at the laboratory condition, but move to laboratory condition to the field. So we have to produce a huge amount of this microbial enriched compost. So we move to a upper scale. So we move to the fermentation, from the fermentation of 100 grams to 5-10 kilograms of substrates. Another difference is the use of the substrates. Until now, I show you a sterile condition. Substrate, it was sterilized. In this picture, you see a non-sterile substrate. And as you can see, we found in non-sterile condition a huge colonization of the substrate by Trichoderma species. In conclusion, we developed a solid state fermentation protocol to enrich digestate with 4 different beneficial Trichoderma species, and we set up a method to quantify the growth of these fungi on a substrate. And this fungus, the goal, the challenge of this technique is the fact that Trichoderma is completely entangled with the substrates. And interestingly, we found that Trichoderma artianum, that is a very common Trichoderma species inside the biofungicides and biofertilizer, is able to induce the germination, the root elongation of both plant species tested. Which are the perspective of this work? We tested and we proved that Trichoderma is able to colonize a non-sterile substrate, is able to win the competition in a non-sterile condition. But what is important, not only Trichoderma, but also the composition and dynamic and also the molecular dialogue between Trichoderma and all the other species inside the substrates. So the future is try to analyze this composition and this functionality of this SYNCOM, this synthetic consortium, on plant resilience, but also in the enhancement of polyphenols, carotenoids, and glucosinolates in horticultural crops. Finally, I would like to thank the other people that work with me in the agri-food environmental microbiology platform, and Cauto that give us the wastes used in this work. And you for your attention.
Thank you, Daniela, for your important contribution. Is there any question from the floor or from the remote side? No question. I have a question. Daniela, which should be the support that the technology can offer, moving the experiment from the laboratory to the field?
Yes, it's a very interesting question because when we think at the field, as I said before from Firat Kuder, they look at nitrogen or other chemical elements present in the soil, but it's also very interesting for us to know which is the effects of microbial community, as also the composition present in the microbial community in the soil. So maybe also sensor or other technical help could be interesting also for us to better shaping.
Please, Anna Maria Bevivino, if you have a question, please.
Yes, of course, it's a very interesting question because the microbial community before solid state fermentation can affect also the ability of Trichoderma to colonize. We didn't analyze the microbial composition at the beginning of this experiment. We used digestate after sterilization process, but also for the security, we store it at -20. So maybe we affect the composition of the microbial community before. So could be another interesting perspective. This is analyzed not only at the end of the process, the microbial community, but also at the beginning.
Thank you, Daniela. We can move to the next presentation.
Yes, me, I have the pleasure to invite on our virtual stage, in this case, Professor Sabrina Sarocco. She's professor in plant pathology at the Department of Agriculture, Food and Environment of the University of Pisa. And she's working on biological control agents against fungal plant pathogens and on transformation of fungal isolates, and also on the identification of new Firmicutes fungi and their employment as source of new antifungal metabolites to be used in crop protection. She's member of the editorial board of PLOS ONE, of the European Journal of Plant Pathology and of the Journal of Plant Disease and Protection. So I have the pleasure. Sabrina, you are connected. Okay, we hear you. So please, floor is yours. Thank you.
Yes, I try to share my screen. So can you see my presentation?
Yes.
Okay. So thank you very much for this invitation. It's a pleasure for me to be here and to talk about a topic I'm very interested in. In the next minutes, I will try to explain how it's possible to use beneficial fungi as a new eco-friendly strategy to control the risk of mycotoxin contamination of food and feed. Just before starting with the presentation, I just like to introduce the global problem, as we have heard at the beginning of this meeting, just because we have a big challenge to face in the next year that consists in the increase of world population by 2050. And to this increase, we have to reply with a great increase in food production of almost the 70%. In this direction, Europe has already moved and is still moving. For example, almost more than 10 years ago, the European Commission invited the member states to find out new sustainable approaches to reduce the use of pesticides, just with the aim to improve human health and to save the environment. In 2015, we signed the 2030 Agenda with its 17 Sustainable Development Goals, among which goal number 1 aims to end hunger, to achieve food security, to improve nutrition, and to promote sustainable agriculture. 2 years ago, we had the European Green Deal with a climate purpose, but at the end of this Green Deal, we have the Farm to Fork strategy. That is a very important strategy with many purposes, just focusing on the reduction of the loss of biodiversity, but most of all, to produce sufficient and safe and nutrient food available for everybody. In this strategy, we have to face 2 important targets. By 2030, both aimed to reduce by 50% the use and risk of chemical and hazardous pesticides. We have also to say that plant disease is a big challenge for crop production, because plant pathogens are the responsible of almost the 30% of total losses in crop yield. In this context, I'm focusing on the purpose to reduce the use of chemical. Biocontrol, that means the use of beneficial microorganisms, including fungi, bacteria, and yeast, represent a valid and eco-friendly alternative to chemical control and also to the use of transgenic plants where these plants are not allowed. Beneficial microorganisms can interact directly with the pathogens by using different mechanisms of action, but they can also be able to reduce plant symptoms and to improve the yield and the quality of the crop. They can be used as the main ingredient of commercial biopesticides, whose market is destined to increase. And you can see from this picture that Europe is more or less in an intermediate position with respect to the market of these products whose ingredients are living organisms. In the big concern of plant disease, of a plant pathogen, mycotoxigenic fungi play an important role because they can affect mostly cereals that represent the staple food of a huge amount of population all over the world. You have to think about the fact that the staple food, the cereals, represent almost the 50% of the diet in Africa. They are responsible of important disease that can affect cereals, but most of all, these mycotoxigenic fungi not only reduce the yield of the crop, but the quality of the crop, because mycotoxins are responsible of contamination of food and feed, especially in post-harvest. Mycotoxins are low molecular weight secondary metabolites. That means that fungi can produce them, but these metabolites are not necessary for the fungal growth, but they have an important role in the ecology of the fungus, especially in regulating the competitive interaction occurring among the producer and other microorganisms, bacteria or fungi present in the environment. It's a problem. Mycotoxin contamination is a problem because mycotoxin can be detected in up to the 25% of commodity. Mycotoxin is a post-harvest problem, but mycotoxigenic fungi can infect the plant during the cropping season, so in the field, so at the pre-harvest stage. In this way, in this context, the use of beneficial fungi to be applied in the field at the pre-harvest stage to counteract somehow mycotoxigenic fungi is a valid strategy to prevent the risk of mycotoxin contamination in post-harvest. In the next slide, I will focus the presentation on two examples concerning two different pathosystems. Aspergillus infection on corn connected with aflatoxin production, and the possibility to use beneficial isolate in the system Fusarium et blight on wheat connected with the trichotition contamination. Here there is a list of some filamentous fungi and yeast actually under research, under investigation, to be applied on wheat and corn with the aim to reduce mycotoxin contamination on the final product. As I told you, beneficial microorganisms, beneficial fungi can directly act against the pathogen by using a different mechanism of action, such as hyperparasitism, antibiosis, the production of lytic enzymes, and hypovirulence, but they can also interact indirectly against the pathogen by competing with the pathogen for nutrients, or they can have a positive effect in terms of plant disease containment, an effect that is mediated by the plant. And this is the typical example of the induction of resistance. In the system Aspergillus corn, the first one I would like to show you, aflatoxin is a big concern for corn production, to the point that more than 100 countries enforced and proposed the regulation to control and to put a limit to aflatoxin levels in feeds and food. Actually, crop resistance and agronomical practices are two approaches, eco-friendly approaches that are used in place of fungicide to reduce Aspergillus, toxigenic Aspergillus infection in plant. But we have also a biological solution because on the market there are already present and available several products to be used for the control of this problem. This product, this biopesticide contains Aspergillus flavus isolate as active ingredient, but these Aspergillus flavus isolate are atoxigenic because nature offer us not only toxigenic Aspergillus strains, but also isolates that are not able to produce mycotoxin, but that are extremely competitive against the toxigenic isolate. In fact, the mechanism of action is the competitive exclusion because these strain, in this case, this biopesticide can be applied in soil at the moment in which the mice is cultivated, that they can compete with the toxigenic strain, reducing them, and at the end, reducing the amount of aflatoxin on mice. It's the sample of the Aflagard that is commercialized in USA. It's the sample of Aflasafe. Aflasafe is the result of a sort of a global project aimed to isolate toxigenic strains from Nigeria and to use this product in the Africa continent just to solve the problem of aflatoxin contamination. Here in Italy, we have another product whose bioactive ingredient is an toxigenic Aspergillus flavus isolate, but at this moment, the registration of this product is only allowed allows its use to mice destinated to feed the production. The second example I would like to talk about, it's the context of Fusarium and blight. It's an example I'm particularly in love with because it's the activity we are doing here in the plant pathology lab in Pisa for more than 10 years. And the problem is a global problem because Fusarium head blight is a disease affecting wheat and other cereals all over the world. It's a complex disease because it's caused by something like 20 different species of Fusarium. And the problem is not only the loss of yield, but also the quality of the kernels, because kernels obtained from plants affected by Fusarium blight can have a very high amount of mycotoxin, mostly trichoterpene, among which the oxinivalenol. The disease cycle of the Fusarium blight is very interesting from a biological point of view because there are two critical steps that are crop residues and spikes atenthesis. Crop residues represent the place where the pathogen can overwinter among two following cropping seasons. So they represent a source of inoculum for the growing plant that show the most susceptible phenological stage at flowering. In fact, the pathogen can infect the spikes, entering the spikes, colonizing the plant, and starting the infection process that can lead to the contamination with the mycotoxin in kernels. The control of Fusarium blight is actually made by different approaches, fungicide, agronomical practices, and also resistance, but no one alone is able to completely control the disease. In this context, the biocontrol strategy can be a successful strategy because we can apply beneficial fungi both on crop residues to reduce the pathogen inoculum, and then on spike to compete with the pathogen for plant infection. It's many years that we are working with the Trichoderma gamsi isolate that is now under development as a future active ingredient of a biopesticide to be used for the control of Fusarium blight. As we have heard in the previous presentation, Trichoderma is a very nice fungal genus because it contains many isolates that can be used as active ingredient of commercial biopesticide. In the specific case, our Trichoderma gamsi isolate is able to grow in presence of a huge amount of DON, or the mycotoxin, that is from an ecological point of view an important feature. It's able to reduce the growth of Fusarium graminearum, that is one of the most important causal agents for Fusarium blight within the Fusarium blight species complex. It can parasitize Fusarium graminearum, and it can reduce the ability of Fusarium graminearum, you can see from this table, the ability of Fusarium graminearum to produce mycotoxin. Is able to compete for natural substrate. We inoculate Trichoderma gamsi in presence of Fusarium graminearum, rice and wheat kernels, and we saw that in presence of Trichoderma, the biomass of Fusarium expressed by nanogram of DNA. It was a real-time experiment. We saw a significant reduction of the biomass of Fusarium graminearum when in presence of trichoderma on both the kernels. And this reduction of biomass was coupled with a significant reduction also of the mycotoxin production. When we moved to wheat straw, the source of inoculum of the pathogen, and we applied the Trichoderma gamsi simultaneously and not simultaneously with Fusarium graminearum, this last just to resemble a more natural situation, we saw that the pathogen reduced its biomass when in presence of trichoderma, in both cases on wheat straw. At the same time, we countered the perithecea developed by Fusarium graminearum, and perithecea are the resting structure of the pathogen, allowing the pathogen to overwinter on wheat straw and to produce ascospores that are the main form of inoculum of spikes during the flowering. Also in this case, perithecea were significantly reduced when in presence of Trichoderma gamsi. Another important feature, Trichoderma gamsi is able to endophytically colonize wheat root, and endophytism is a prerequisite for the induction of plant defense response, from the induction of resistance in plant. So we demonstrated that this strain is able to colonize endophytically roots, to colonize endophytically spike tissues, but also to modulate an overexpression of several genes connected with the plant defense. When we moved in field, we observed a significant reduction of the disease index and the disease severity occurring on wheat inoculated with Trichoderma gamsi. But this experiment was very important because by a metagenomic approach, we could demonstrate that the application of Trichoderma gamsi on crop residues, on spikes and on spikes did not modify the natural bacterial and fungal population present naturally occurring on wheat straw, on spikes, and on kernels after the harvest. And if you remind, one of the purpose of the farm to fork strategy, that is to reduce the loss of biodiversity, this is a good point in favor of the application of this isolate in field. Just because we like to enter in details of what happens between two fungi, we made a transcriptomic analysis just to verify the crosstalk occurring between Trichoderma gamsi and Fusarium and graminearum. We make a transcriptomic analysis aimed to evaluate the differentially expressed genes in Trichoderma and in Fusarium when we were put one in front of the other, when they were able to chat somehow. And we noticed that while Trichoderma gamsi maintained a stealth mode, that means he down-regulated the greatest part of its gene, Fusarium graminearum was in a buzzing mode because he upregulated almost more than half of its the expression of half its gene. Among these, we notice an overexpression of the so-called killer toxin-like protein that we had finished just a few days ago a paper about this, that probably they are involved in the competitive interaction of this Fusarium with something else that can compete with them, in this case with the beneficial isolate. Last information I would like to give you, we sequenced the genome of this isolate, and we noticed that in this genome is present a 3,5 gene. This 3,5 gene is a gene encoding for a terpene synthesis. We have heard about talk David Colling about this class of metabolites, but the most alerting things is that the 3,5 genes is also the first gene that is expressed in fusarium when trichotichens are produced. There are trichoderma isolate, trichoderma species able to produce trichotescens, trichoderma rhabdinaceae and trichoderma brevicompactum, but we made a phylogenetic analysis and we saw that this we called at the moment putative 3,5 cluster gene in Trichoderma gamsi seems to be present, to be spread among Trichoderma genus, but in our cases we demonstrated that even if the gene is upregulated when Trichoderma interact with plant, it is not responsible for the production of any trichotescens. So because it's important to know everything about an isolate when you want to move to an industrial scale and to develop it as a principal ingredient of biopesticide, you have to exclude, of course, that this isolate can be harmful for the environment and for the humans. So we are quite sure that this isolate could be another little strategy to contain this important disease, fusarium blight, but also the risk of mycotoxin contamination. Just to conclude my presentation, we have to reply to this global request of sustainability. We have to do it also in agriculture to increase yield and quality production, because we have to face the problem of the increasing population. Biological control, the use of beneficial organism is a tool to reply to this request, but, and this is well explained in a review we recently published also with David Colling, we have to move from, when we use biopesticide, we have to move from niche market to a broad, not only industrialized farming, but also to a broad way of thinking. We have to accept this new product because they are the future for obtaining a safe food. So I hope to have illustrated you what we are doing, which is the progress, which is the already existing solution. And I thank you for your attention.
Thank you very much for your interesting talk. I don't see any question from the remote audience. I don't know in the room. In case I have one quick, because we don't have a lot of time, but I guess it is quick. I'm from engineering, so my question is related to which will be your dream for having a measurement on the field for the effectiveness of your treatments? We are ready to do something. If it is not, just let me know, but I'm curious.
My dream or the scientific dream?
your infinite budget, go ahead.
Okay, no, my dream for sure is at a more wider level is to move what we reach in the lab to the real life, because of course we are academic, so we are in the lab, we enjoy working. The dream is that really the use of beneficial organism, not only alone as a single ingredient, but in a wider perspective as a community, as consortium. We have heard the previous presentation that is aimed to reach this. The dream is that this kind of organism that normally has a positive effect in nature can be used for trying or to give a contribution to solve the problem of feeding the world, and also to produce safe food. So from an industrial point of view, we are receiving a great attention because the legislative situation is calling also the companies to produce more sustainable product. And we have to solve the problem of the reduction of the 50% reduction of chemical in the next less than 10 years now. So my dream is that what I've told you now could be really reality in the practical life.
Thank you very much. I thank you again for your speech, and I move to the next one. That is Dr. Victor Grimblatt from Synopsys Chile. Chile is a managing director of this company. We are not in the microelectronics audience, but Synopsys is one of the best companies for microelectronic design and tools in the world. In fact, Victor is an high-tech executive with a broad experience in marketing, sales, design, development, and production, as well, of course, experience working in multinational companies. But he's here in particular because during his PhD, he did and designed the first, we can say, but at least one of the first microchips, microelectronic chips for precision agriculture. He is a member of the board of governors of the IEEE Circuits and Systems Society in the advisory board of Talento Digital. He was general co-chair, you can imagine who was the other one, at Foodcast 2021, and is president of a special interest group on the agri-food electronics of the Circuits and Systems Society of IEEE. So it's my pleasure, Victor, the floor is yours.
Okay, thank you very much. Good morning, everyone. I hope you are listening me well. I will try to share my...
Perfectly, Victor.
Thank you.
It's very early in the morning, so I hope that you had a very good coffee today.
No, that's okay. Just 6:30. And it was very interesting to listen to the presentation.
Okay, please share your slides.
Yes.
Okay, they're coming.
That.
Yes.
Now I'm putting presentation mode.
Yes.
Okay, this is
great.
So, thank you. So thanks for the invitation. It is true, it is early in the morning, but that's okay. That's not a problem. So I'm trying to tell a little what I did during my research about to increase the soil productivity in a sustainable way. And I'm not sure that this is the dream of everyone that is working in agriculture, but I will try to at least to address some part of this dream. So that started with my own personal experience, putting some plants into the land, some tomatoes, some lettuce, and I started irrigating them. And of course, after some irrigation, I got, sorry, we're having a trouble there. So I irrigate and I continue irrigating, and some of my plants die and some others succeed. So I was wondering what is happening here, because for me, in my personal knowledge, I'm not an agronomist, I'm just electronics. So I was thinking that putting water is enough. So I conclude that the plants need more than water. So I say, okay, we need to investigate that. And on the other hand, one agronomist that is becoming one of my friends told me we are very far away from smart irrigation. So I say, it's so easy. We put some sensors. No, we are very far from that. And also, the other thing I learned, that probably you all know, that tomatoes, lettuce, they're all different. It's not one vegetable that we can find, but there is a lot of different vegetables, and all of them have different requirements. And what about fruits? Because we talk about vegetables, tomatoes, and so on, but what about fruit? So after that, I started researching, and I will divide my presentation in the problem. After we'll go to the IoT, which is Internet of Things, and what can be applied, and finally some experiments I did applying that to horticulture and fruit growing. So the problem I want to summarize, I have put the main part of the problem, and I will summarize that, is the first one is 1/3 of global greenhouse gas emission is caused by agriculture, forestry, and change of land use. So we always talk about this problem of the climate change that is affecting agriculture, but on the other hand, agriculture is affecting the climate change. And here you see some numbers coming from the Food and Agriculture Organization of the United Nations showing the impact produced by the agriculture. And for example, the nitrous oxide from fertilized soil is 17%. It's a lot. The land conversion, 47%. So we have to start thinking on how we can work on that. The other big problem I see is the fresh water. And we tend to listen and to think that the problem of the fresh water is coming from industry or from the domestic usage. And everyone is telling us, yes, you need to decrease the time you are using the shower. You need to, but if you look at those numbers, the agriculture is the heaviest consumer of our planet after fresh water. And probably an important part of that is wasted. And we see that Europe is, is very well balanced between the three consumers of fresh water, the agriculture, the domestic, and the industry. But if you go to North America or even to Asia, you see that more than 70% is used by agriculture. So we need to decrease that, and 70% of blue water withdrawal from water course and groundwater are destined to agriculture. And the agricultural water demand is estimated to increase by 19% by 2050. At the same time that we need to produce more food for 2050. And another problem that in general we don't analyze too much is the farms, how they are distributed. 80% of farms operate on less than 2 hectares of land. So we are talking very small farms, about 500 million farms smaller than 2 hectares in the poorest country, where we use a lot of fresh water, by the way. 70% of land is operated by farms smaller than 5 hectares. So what is the implication of that? Farms are very, very far from technology. They're not adopting technology. What are the reasons? They are saying too expensive. When we think on someone that has 2 hectares of land, that is a person that is produced maybe its own consumption of vegetable and fruits, maybe selling some part of that, but not too much. It's hard to use. Technology is hard. It's always a mystery how it works. They don't have digital skills. The farmers, they don't have digital skills. I'm talking about the experience in my country. Farmers, the small farmers, they use Facebook, they use WhatsApp, they use a cell phone. But from there, starting to tell them, yeah, you will have the result of the humidity of your soil here, that is a little more complicated. And the communication availability and cost as well is a big problem. In some part of in a lot of part of the world, in the countryside, there is no communication, there is no internet, there is no 3G, there is no 4G or 5G, there is 0G. So the communication is also a problem. Some 5 key takes away from United Nations, okay, that humans have already transformed more than 70% of the Earthland's area, food system responsible for 80% of deforestation. So we see that we are facing a big problem. The world faces tough choice between protecting and restoring land and business as usual, because we're still working in the same way we were working in the, I don't know, in the last 20 years. We have had some changes using mechanization, but we have to do much more. And the previous presentation, we listened about this sustainable objective from the United Nations. And what I like is to also show this donut economic and planetary boundaries. So the donut economy that was created by Kate Raworth, who's an economist in 2012, say that the old economy has to be based in a donut having two borders. The internal border, what she called the social foundation, has to be based on the United Nations sustainable objective. And the outside border, the ecological ceiling, has to be based on the planetary boundaries defined by Johan Roxon from the Stockholm Resilience Center in 2009. And you see that you have things like biodiversity loss, land conversion, nitrogen, and phosphorus loading, climate change. And it is important to see that almost all of these parameters that are in red have been affected by agriculture. So agriculture is one of the ones that is really creating, it's a big responsibility of the climate change, and we have to take that in account, and we have to do something. So we have a lot of problems. We have a problem that has a lot of requirements, several dimensions, more population to feed, less arable land, climate change, less fresh water, ecological footprint. But on the other hand, we need system that has to be low cost, because We have small farmer, low power. We don't want to put batteries everywhere, easy to use and able to communicate. So we have a big problem. Agricultural dilemma, increased productivity versus planet boundaries. So IoT, how to solve it. And this is why we say the IoT Internet of Things to the rescue. So do we know how plants grow? I will not go into details of that, I suppose you know, but it's a very complex system having different kind of parameters that could be from the plant, from the soil, from the environment. And we can try to find a model, but there is no model today of growth. So this is related to the soil, the different things we can measure or we can try to measure, the texture, the water, the nutrients, the salinity, etc. This is what we can try to measure from the environment. And if we look the parameters to consider for the soil, we see that a lot of them are fully dynamic, meaning that they will change very often, and we cannot control how they change because the water is based on irrigation and the percolation and all that, so it will change. And can we artificially modify? Yes. The nutrients as well. The pH, yes, we can also modify the temperature of the soil as well. But there are some of them that we cannot modify on the environment. For example, the light. Light, unless we are in a greenhouse, but if we are outside, we cannot modify. The weather cannot be artificially modifiable, etc. And one that is important, we say the salinity can be modified, but it is very hard to change the salinity of the soil. We have to clean the soil, we have to wash the soil. Can we measure all of them? Unfortunately, it's very hard, so we are going to go to IoT. So the main concept, what is IoT? The Internet of Things is the network of physical objects or things embedded with electronics, software, sensors, and network connectivity, which enable this object to collect and exchange that data. So in fact, it is the connection between the real world with the digital world. When I say the real world, the world is analog, it's not digital. So we have things that are moving, we have things that we sense a lot of things, and we need to convert that to digital so we can process. And this is the Internet of Things. What could be a thing? Anything, a cow, a plant, a building, a tree, a car, a container, a boat, a ship, a luggage, everything. What are the main markets for IoT? We have the smart home, the industrial IoT, the connected vehicle, the health and fitness. And you see the agriculture, we talk a lot about IoT for agriculture, but there is very few applications that already exist because in general, the industry is mostly working on those things that are where we have a lot of customers. We know as an industry, technological industry, that the customer in the field of agriculture, they are very small. So they will not be able to buy big systems. So research on one on the other. The architecture of an IoT system is you have the devices here, which are the sensor connected to something to send information. You have the gateway that is concentrating several sensors. And there we arrive to the network and we send that to a data center where the data is processed and we went back to provide information. So going to that part of the system, the devices, how to connect a thing to internet, because how to connect a cow or a lettuce to internet, we have what we call a node, the edge, that will transmit information through a radio link to a concentrator or a gateway that will transmit information to the network link through internet to the server. What is the node? The node is a device, electronic device that have and security. It is important that we take in account security, the network access. We have sensor and actuator, sensor that will sense information, actuator that will provide some orders to valves, engines, or whatever we have in the field. We have the digital analysis, all that. We have the digital process. We have the communication, radio frequency, and we have the power supply, how to power this electronic, because we need to power the electronic. How to connect, the connection time and how to choose, because the connection will depend. We have learned a lot of, we have heard a lot of names, ZigBee, Bluetooth, Wi-Fi, 2G, 3G, 4G, 5G, etc. But how to be sure that we are using the right one, it will depend on the amount of information we want to send and also the distance in which this information has to be sent. For agriculture, we use the most uses is LPWAN, low power weight area, and we have Sigfox and LoRa as the leaders on that. And the reason is that we want to send very few bits of information because we want to send a number, the temperature or the humidity, very far. We have from time to time to go to 1 kilometer, 3 kilometers, or even 10 kilometers. Edge computing is something very important, refers to the computation and analysis of data on the distributed device instead of the cloud. So we are doing more and more going to edge computing, which means that the node, the edge I was talking, will be smarter. And according to Gartner, by 2025, 75% of data processing will move to the edge, up from 10% in 2018. So that implies we need to do a very low cost system because we are going to have a lot of these small devices put in the field every 10 meters, 20 meters, I don't know, 20 per hectare or whatever. So we will have to put all that and start processing the data we are getting. So
Victor,
the edge
please, because of
yes.
time constraints,
okay.
You have 2-3 minutes. So go, please.
Okay,
thank you. Sorry.
So the architecture of the edge, you see it's coming back. So main requirements, I will try to go there. So what we have done so far is, okay. Oh. Okay, so we did some experiments. We measured soil moisture with a resistive sensor, and it was a 4-day experiment. The objective was to analyze the interrelation of parameters, and this is the results we are getting. So the soil temperature is following this kind of waveform, and the soil moisture was coming from wet to dry situation, mostly a couple of days. So now we have to analyze all those points. Second example will be also to measure. We use 2 kinds of sensors for humidity, resistive, and capacity. And we were also measuring the soil temperature and the environment temperature. That is the analysis we are doing. Some more numbers, bell pepper, soil and environment temperature, the moisture as well, and some conclusion to conclude on that. So what are the topics that caught my attention and keep me awake? The lack of low power and low cost sensor for some of the parameters influencing plant growth and health, how we can generate this equation and model of plant growth and the interrelation between the parameters, better understanding of plant growth for better IoT system, and agriculture provides our food. Population is growing, so more food is needed. Arable land is decreasing. Need to improve the soil skills. Precision agriculture and IoT we consider is a key for that. Machine learning will allow the IoT system to work independently. Artificial intelligence will provide algorithms to improve the yield. And of course, we need to work close to the agronomists because we have to very well understand the problem so we can try to provide some solution. Sorry for the delay. Thank you very much for your attention. I'm available for any question you could have.
No worries, and thank you for your presentation, Victor. I don't see questions from the cloud here. Emilio has a question. Please, Emilio.
Yes, thank you, Danilo. Thank you, Victor. Very interesting overview of the technology possibility. And one of the main problems you focused was the energy. What do you think about the possibility of using techniques such as power harvesting?
Yeah, this is a very good question. It is possible to use energy harvesting. The main question will be now, do we want to have a battery or not there? Because energy harvesting can go directly to power the system. And also we can have a battery that could be could be recharged through that energy harvesting. If we want to avoid batteries, energy harvesting will be the solution. However, we have to do a very good power management because there will be moments where we are using a lot of power. So we have to balance that. It is possible to use it. It is a very good solution. I have even read a paper from some people using the energy coming from leaves, but the system we are going to produce have to be very well balanced in energy so the harvesting will be enough.
Thank you, Victor. I don't see other questions. Thank you again for your presentation. And now, last but not least, now we have here in presence Professor Daniela Uberti, who is full professor of pharmacology here, University of Brescia. She's working on proteomics and redox proteomics and nutraceutical research, and also on electrochemical sensor for biomarker detection. She is a co-founder of a university spin-off named Diadem. She is today visiting scientist at the National Institute of Biological Science at the Scientific Park of Beijing in China. And last but not least, absolutely, she's director of the NutriFund for Health Center, focused here in Brescia on functional food and nutraceutical. So, Daniela, my pleasure is to give you the floor.
Thank you, Danilo, for the nice presentation. Let me just thank all of you, especially who is virtually attending, that are still there despite the lunchtime. So I will try to be brief. My presentation, I want to give you another point of view. We are all aware that there's a close correlation between diet and health. And fruit, vegetable, all grains are recommended in Mediterranean diet because are rich of bioactive compound. This bioactive compound is endowed of peculiar properties and that have an impact on health. And don't forget that if you get aware the population in consuming healthy food translate in less public spending. When we speak about a bioactive compound, we are talking about secondary metabolites. And the plant synthesizes secondary metabolites to interact and protect itself from the surrounding environment. And we take advantage from their behavior for our purpose. So as we also heard today, we try to get in place a new strategy for enriches veggie with this or that bioactive compound for our health. The final aim, what is the final aim? The final aim is to obtain a food qualified as functional. Functional because according with its definition, it's a food rich on the enrich of bioactive compounds that should have a health benefit. But with the view that in the near future, functional food could really become a systemic approach for maintaining health and even cure chronic disease, it is imperative to adopt a multi-step process that gets forward a rigorous and systemic experimental approach, and should be interdisciplinary, should take or should need an interdisciplinary expertise, starting from the agriculture and the study of the plant and the characterization of bioactive compound profile, but also the expertise from chemistry and pharmacologists for characterize and study the bioavailability, the mechanism of action, and also expertise from clinicians, because we need to demonstrate the effectiveness in the human being. Having in mind this interdisciplinary approach, we recently founded the NutriFund for Health Center and focus on the research of nutraceutical and functional food. For my professional setting, I see at least two main challenges the functional food have to overcome. The first is the standardization of the product in order to give always reproducibility and reliability results. And the second main challenge is to optimize the production in order to fit in a eco-friendly process. This means reduce water consumption, avoid pesticide, and recycling, and valorize byproduct waste. Now, today, I want to show you a new edible ecosystem product developed by a local startup, Cibo-C, and that see the collaboration of different institutions, Spallanzani Institute and University of Brescia, in order to characterize some aspect, scientific aspect of this functional food. The main question is, can we define this edible ecosystem a functional food? We are trying to answer. So there's a great interest in microgreens, and microgreens differ a lot from mature form for the content of bioactive compound. Microgreens are a huge sources of minerals, vitamins, bioactive compound. So starting from the microgreen concept, it has been developed Cibo-C, the edible ecosystem. The first step has been the choice of the seed and of the species to culture, and the company focused on the brassica, and they choose these three species of brassica. And why brassica? Brassica, because brassica is rich of glucosinolate, and glucosinolate actually is inactive, but they give after the activation by a myrosinase enzyme to an active compound that is sulforaphane. Sulforaphane is widely studied, and there's a lot of literature about sulforaphane and especially cancer, and the beneficial effect of sulforaphane are mainly due to its antioxidant and anti-oxidative properties. So I show you just the step of the production of this ecosystem. There's the matrix is prepared and sterilized and filled filled in the well box, and the seed previously primed in water and treated with hypochlorite are seeded, and then the box is closed with a porous plastic film that allow the gas exchange and avoid the contamination. The plant is grown in a vertical farming under a LED grow light for 14 days, and then it can be eaten just as it is, or just only open the box. Taking in mind that this product, this ecosystem is completely edible without any waste. So then with the Spallanzani Institute, we characterized the polyphenol content, the total polyphenol content at a different stage of growth, just after seeding, after 1 week, and after 4 weeks. And as you can see, there's an increase of 2-fold of total polyphenol in the ecosystem after 1 week, that remains stable at least until 4 weeks. We in Brescia measure with HPLC the amount of glucoraphanin and sulforaphane, and this is the amount we found that is in any case a good amount comparing with the mature brassica plant. Then the antioxidant activity has been measured, and this with the DPPH test, and we can see an increase of polyphenol content is correspond to an increase of the free radical scavenger activity. The plant ecosystem, the matrix that is edible, give the opportunity to be added with additional components, and we choose 2 formulations. And these 2 formulations, it's important to underline that these 2 formulations comes from circular economy. And one is the Hematococcus pluviale, and it has been chosen because are a naturally sources of astaxanthin, and the extract of grape leaves rich of polyphenol, and especially in resveratrol. And when we add these 2 formulations to the matrix, what we realize soon is that is not just a simply fortification, but the constituent of the formulation dynamically interact with the growth of the plant. And in fact, as you can see, there's a different morphology of the roots using adding the different formulation. This means that we obtain not a fortified ecosystem with, but a new ecosystem, a new product with a potential new properties. And so we test the antioxidant activity of the ecosystem added with the two formulation. At the Spallanzani, as we conducted ORAC test, ORAC assay, as you can see in the polyphenol enriched grape extract show a higher antioxidant anti-activity compared to the original one, and also Hematococcus pluviali show an increase in antioxidant activity. And when I speak about antioxidant activity, in this case is a free radical scavenger capacity. I want just that you focus on the Hematococcus pluviali at the T1 that here show is not so high results with ORAC test. Why I told you to focus on these particular results, because I want to explain what for us pharmacologists is quite clear, but not for all, that there is a differences between free scavenger activity and the capability to protect against oxidative damage in a cellular context. In fact, the cells is a door of a system that allow it to defend itself from free radical and oxidative stress. And this system involves the activation of intracellular pathways, quite complicated intracellular pathway. And I want to, and so the question is, are these products also able to improve the capability of the cells to react and to respond to an oxidative damage? So we measure the activity of an enzyme catalase. Catalase is an enzyme present in our body and that is able to detoxify from hydrogen peroxide. And as you can see, the addition of Hematococcus pluviali and the grape leaves extract are able to activate the enzyme and to render it more prone to react with H2O2 and detoxify it and convert it to water. And this is done using the T1, the T1 sample. And here, even the Hematococcus pluviali extract did not show an ORAC result so interesting. Anyway, it has a very interesting antioxidant activity in a cellular system. Then we test also the capability to prevent or to counteract an oxidative insult elicited with H2O2, and the original product means the ecosystem without the addition of formulation of the two products, the two ecosystem added with Hematococcus fluviale or the polyphenol extract, showed a protective effect against the oxidative damage. And you can better see in this in this graph the percentage of protection that is around over 30% for the two formulation. So of course, there's a lot of work to do yet, but at least these data highlight two important things. The first is the important to use different experimental approach to demonstrate the biological activity. And the second, very important, is the versatility of the matrix. Means that this experiment underline how the matrix can be personalized, tailored with different formulation for the purpose we have in mind. So there's many possibility, can give very many possibility to obtain a new product. I leave you returning to the definition of functional food that gave EFSA and Food Center research. First of all, a functional food can be defined functional if you demonstrate, experimental demonstrate its beneficial effect. So it's need a rigorous experimental approach to study the mechanism of action of this product. Of course, it's important also to demonstrate beneficial effect in a trial study, because we have to demonstrate, to demonstrate that this functional food clinically provide and clinically proven and documented health benefit for the prevention and management and treatment of chronic disease. And let me thank the colleague who contribute to this research, Cibo Ci and his founder, Marino Novello, but also the Spallanzani Institute with Silvia Cenadelli, Graziella Bongini and Roberto Puglisi, and my lab, with Giulia Abate, who is the coordinator of the lab, and also the new born center, NutriFan for Health, with the colleague who joined me in this adventure of the center. Thank you very much.
Thanks to you, Daniela, and compliments for the very good work. And also, before moving to the conclusion, me personally, I want to thank and give my compliments to the colleagues here in Brescia for the hard work and the great work that you did for organizing this workshop, and also supported by MZ Events. So thank you so much for inviting me to be here, and my compliments for a fantastic job. So now, Emanuela Gobbi, please, I pass you for closing remarks.
I will be very short, very brief and synthetic because it's so late. Just to tell that I hope you enjoyed the morning. I hope this event that was intended to be a concrete action for sharing the One Health concepts by recruiting different social communities work and in making these communities more conscious and perceptive. The goal, the success of this goal requires, I think, an interconnection, a partnership, a network among universities, but also among universities and institutions such as FAO or the global or the institution that many universities are creating, as the one directed by Professor Scetti. And I hope that Brescia and the University of Brescia demonstrate to be active protagonists of this networking. Thank you all for your participation and attention and patience. Goodbye.